Hello, 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 everybody. Welcome to the stream. Welcome to you all. It's been a while. Hello, hello. Everybody, say hello to my special guest. Special guest, please introduce yourself.
Hi, I'm Myelin Aikman. I do a channel on psychology, anime, statistics, and that sort of thing.
Yeah, so I've had a bit of success in inviting VTubers on my channel where I get to share with them all the things that I truly love about biology, and I want to be able to do this with a bunch of VTubers sort of all over the academic sphere, just because I think biology is so central to our lived experience that almost any academic will have some sort of overlap with something in biology. And so we went back and forth sharing a couple of papers, and I thought this would actually be a really interesting paper to share with someone who does psychology, because this paper has implications for how the microbiome connects to how we behave, and that has been a very hot topic as of late. But first, I just want to say hello to all of those of you who are in stream today. My mouth keeps on getting tangled up. Like I didn't stream last week, and now I feel like I've lost some of it. So I'll start off by just saying hello to chat. Hello, Cool Guy and Spepic and Alice. Hello, Lunamar. Hello, welcome. The Wolf. Welcome. Marlin. Brian. Endless Space. Hello, welcome for the first time. Yes. This is going to be a great conversation to have. I think this paper's super interesting.
Should definitely be fun.
Yeah. Spoilers. Myelin and I have talked at length about this paper behind the scenes, so much so that I kept on having to be like, save it for the stream, like let's not like throw everything we have out there just in the behind the scenes conversation. But this paper is going to be very interesting for the both of us to discuss, because you have an outside perspective, which I think is going to be very fun. Hello, Lil. Not going to finish that username just for YouTube community guidelines sake. I don't know if I said hi to you yet, Luke, but hello. It's going great. I'm very excited to be streaming right now. Myelin, I just want to ask you sort of, or not ask, but rather just, we have similar backgrounds, I would say, given our professional lives include teaching. And I think what's interesting about that is that we both took sort of different, you and I have different perspectives on this paper, and I think the internal teacher in both of us have really allowed us to communicate that difference in perspective in a respectful manner, which I totally appreciate. But when you pointed certain things out about this paper, I was like, oh God, the psychologists are going to believe we live like this. And I got really self-conscious out of nowhere.
Sorry, that was not my intent.
So we're going to get into why in just a little bit. But that's just the teaser for you out there, that I certainly learned a lot more about how psychologists think than I expected. Because here I thought, I'm just going to share this biology paper with a psychologist, and we're just going to have a conversation. And you know, this biology paper will challenge them, and it'll be fun to like watch them struggle. When in reality, I don't think you struggled that much with the paper at all. In fact, you had pointed things out about the paper that made me question how well I understood the paper, which was a bit of neat role reversal. Okay.
Well, I mean, I learned a lot from you, so I learned a lot from this paper in general. It was really fun.
Yeah. All right. So let's talk a little bit about why we are gathered here on a Friday evening to watch two science VTubers stream. Today we are covering the paper Diet and Exercise Orthogonally Alter the Gut Microbiome and Reveal Independent Associations with Anxiety and Cognition. A lot of fancy words that we'll get into later. This is by Kong et al. In 2014 in the journal Molecular Neurodegeneration. So right off the bat, this is an older paper, although I shrivel to dust every time I think about the fact that 2014 was 10 years ago. So yes, this does count as an older paper.
Don't say that.
I
mean, I get it, but
it's a little shocking, especially because this information is so, I feel like Gen Z might be already very accepting of the conclusions here. But, you know, when this paper was published, this was like new, exciting stuff. This idea of a gut-brain axis, that your stomach, you know, the context of your gut, the microbes that live there can influence how you feel and behave. That was very new back then. And I picked this paper not only because it's open access, which is great, so I can show the actual data, but also because this appears to be a pretty important paper. It's got 400 citations, which is a good number of citations for a paper to have. But I don't want to come across as though this is like a defining, excellent research paper that's rock solid and you can't poke holes in it. What I would say is that this paper ignited, I think, the imagination of a lot of scientists, and we're going to get to kind of see what the sparks of that imagination may have looked like back in 2014. So this is, I think, more of a historic kind of paper.
Mm-hmm.
All right, so let's go ahead and get right into it. The first thing that I want to make very clear to all of you watching, the microbiome field is filled with grifters, people who want to separate you from your hard-earned cash. So I want to preface this by saying, do not take any action based on what you learn without approval of your doctor. The thing about microbiome research is that it's almost like created in a lab to be something that grifters can take massive advantage of, right? It's very, very important. The microbiome is extremely important to human health. It's very easy to simplify. You got, bacteria and fungi live in you, and the happier they are, the happier you are. That's a very simple way to put it. It's hard to verify certain claims. So, experimentally, it's quite difficult to do this kind of research. It's hard to fully understand. As we're going to get into this paper, we're going to see that it's kind of hard to take anything away if you really, really think deeply about the conclusions. And of course, the mother lode of grifters out there is the fact that this can be sold as a supplement. So there are going to be people that try to sell you things that promise all sorts of things. Be careful what you believe on the internet. Do you have anything to say to that, Myelin, considering you do study conspiracy theory, correct?
Yeah. I think I was telling my students the other day, one of the things to look out for when you are trying to figure out whether you can believe some information is, are they trying to sell you something? A lot of misinformation is used to sell you ideas, of course, but also products like supplements. And I can think of one particular person who's famous for spreading misinformation who sells supplements in particular.
Yes, we will not mention their name, but they have made
Nope.
quite a bit of money doing so. But yeah, so just be aware that I'm sure that if you're tuning in, that you are at least a little interested in the microbiome. It's still in the infancy of research. There are approved probiotic treatments out there for very, very specific diseases, and I say diseases. So anybody trying to sell you a probiotic just to say, "Oh, this will make you feel better," if it's not medicine, then it's not approved by the FDA, and FDA approval is important for things that count as medicine. So this could be an entire stream on its own. We're not going to get too deep into it, but just at the top of the stream, just because I had a couple of comments asking, what we can do about our microbiome. And my answer for you is, I have no idea. And this paper is certainly not going to give you a better idea. So I hope we're all on the same page about what it is we're actually here to learn. All right? All right. Here we go. So there's this idea out there called the gut-brain axis. This idea that the brain and the microbiome are intimately connected, that things that happen in the microbiome can influence the brain, and things that happen in the brain can influence the microbiome, right? So how brain/behavior can affect the microbiome is through immunity. So your brain can, control center of the body, can help elicit immune reactions that can certainly change the population of microbes that you can find in the gut. These kinds of hormonal signaling cascades can also increase or decrease permeability of the gut lining, which can also affect the gut microbiome. The body can also create mucus, and mucus, as you will find out in the video that I will be releasing hopefully by the end of October, is a really important molecule that can help tame and regulate the microbiome and other hormonal signaling cascades, right? So the brain can affect the microbiome, but as we are also learning, the microbiome can affect the brain. So lots of microbes that inhabit your gut can release metabolites and neurotransmitters and immune modulators that can have downstream effects and affect how you feel and how you behave. So this is something that scientists are pretty sure of. It's been experimentally verified. The question is, what do we do about it? And that relates back to the whole grifting idea. That part's not entirely clear, but this connection is clear, right?
Mm-hmm.
So This paper seeks to ask the question, okay, so the brain and the gut lie on an axis where they can affect each other. What about diet and exercise? So you could hypothesize that anything that introduces new molecules into the microbiome or new pathways in the brain can affect the microbiome, right? That just makes sense. It intuitively makes sense, right? If you eat a lot of pizza, then your gut microbiome might look a little different than someone who eats a lot of vegetables. You can just intuit that, although of course you would need to experimentally verify that. So back in 2014, these scientists wanted to figure out how these four things are related to one another. So chat, I want to take a little bit of time to hypothesize the nature of these relationships. So imagine there are these four corners and you could draw arrows between any two of these in any direction. Let's just get some ideas as to how diet relates to the brain, or how diet relates to the gut, exercise to diet, exercise to brain, etc., etc., etc. So, what can we hypothesize about these relationships, right? Brain/behavior control whether or not you diet or exercise, certainly. So
Ooh.
there are top-down decisions that you can make that can affect whether or not you choose to exercise. That is, again, of course, if we believe in this idea of free will, which is I'm maybe not the VTuber to talk about free will, but certainly that is one line that we could draw. What else? Perhaps certain microbes will prefer different sources of foods that you eat. Certainly so. In fact, when I was in graduate school, we had a super interesting laboratory that was using cheese as a model for the microbiome. So yeah, it was crazy. They would like culture cheese, and they were investigating the dynamics of microbial colonization. So you can imagine like an uninhabited island is first colonized by like ferns, which are then eaten by simple animals that die and decay that provide complex nutrients for trees. And they were using cheese as a way to get a clean environment and then just see how different populations of bacteria mature over time. So certainly the local metabolic or food environment can definitely, you could imagine, change the microbiome. What else do we have? Exercise affect diet, since you will eat more if you are tired. Yeah, and that effect might go through the brain, right? So if you exercise, you might be rewiring your brain. Your brain might be sending out more hunger signals. Your hunger signals might tell you to eat more. You could certainly draw lines through that. What about the virome? Since the virome affects the microbiome, yes, certainly. You could imagine that the virome would shift naturally with the different types of bacteria that exist within the gut. That certainly makes sense. The intestines can have subtle nervous stimulants that drive particular cravings. Exactly, yeah. So, the gut has nerves in it as well, and those nerves can talk to the brain. These are all really great connections. So here's what I came up with, okay? You can draw this line. So certain diets can encourage certain microbial growth. Diet, if you have sufficient nutritional intake, you can nourish the brain, healthier brain. Diet goes to exercise. Sufficient caloric value gives you accessible energy, which enables you to exercise. The brain affects diet through dietary choice, right? Like the foods that you seek out. Brain affects exercise through the adaptation of behavior. Brain affects gut microbiome through signaling pathways, as we saw earlier. Exercise can affect the brain through feel-good chemicals. Exercise can affect diet by changes in appetite once you start exercising. Exercise could affect the microbiome through a mechanism I have no idea. And that's part of the research question that this paper poses. And the gut microbiome, as we saw before, can affect the brain through signaling and maybe affect diet through some other mechanism that maybe doesn't have to go through the brain. Who knows? So what I want you to maybe appreciate here is how complicated the potential linkages between these four ideas actually are. And this is why I actually tend to stray away from research papers that look at stuff like this, because I'm a simple neutrophil. I see causation in an experiment, and that's what I like. Correlation I am deathly afraid of, because correlation it's just not satisfying to me. So I took this opportunity to chat with someone who deals with correlation every day.
Yay!
So these connections are messy and complicated, so who better to share that with a person who studies the messiest biological system to ever exist, right? People. Right?
That's me.
Yeah, so chatting behind the scenes, I was like, and we're getting a little ahead of ourselves, but just as a teaser, you were like, actually, these like correlations look really good. Like the effect sizes are good. And I was like, what are you talking about?
Yeah, it's all relative, isn't it?
Yeah, yeah. It is all relative. So that's like the first striking difference in perspective where like bad or small effect sizes in our side, not bad, I shouldn't say bad, but like low magnitude effect sizes on our end or low correlative values. One man's questionable p-value is another man's research paper.
Indeed. I mean, you're explaining 20% of the variance on a lot of these. That's pretty darn good as far as I'm concerned.
Absolutely blows my mind. Correlation is not causation. Yes, exactly. So that is why I prefer experiments that show causation. I tend to not talk about correlation because it doesn't make for as good of. I would have to do a lot of please interpret this with caution throughout the entire stream. And I typically don't like to do that. But that's like the entire field of psychology. So
oh yes, that is, please interpret with this as caution is my bread and butter.
Yes.
So I will do my usual corollary. Correlation's not causation, but if there is causation, there will be correlation.
That is true. Correlation is, in fact, correlated with causation.
This is true. It's not a perfect correlation, certainly, but it does exist.
Yeah. Could exercise affect how the immune system can regulate the virome microbiome with effects? What kind of viruses? Yeah, certainly. What is the virome? So the microbiome describes all of the little bacteria / yeast / fungi / archaea and one more big class of microbe I'm forgetting. Toxo, what are you? T. gondii. What is your species, your order? What are multicellular microbes? The virome specifically refers to the types of viruses that you can find in your gut.
Oh,
yeah.
I knew I'd learn new things today. I've learned a new bit of vocabulary. Yeah. Thank you.
So whenever you see "ome" at the end of something, it refers to like the complete collection. Not eukaryotes. Dang, it's gonna bother me if I don't get this. Protists. Yes, protists. Multicellular eukaryotic microbes. All right. So this paper takes a look at the effect of diet and exercise on the gut and behavior. So making no assumptions, we're going to have experiments that influence diet and influence exercise, and we're going to measure things in the brain and measure things in the microbiome. So to measure effects on the brain, we're going to be, not we, but the authors are going to be measuring anxiety and a little bit of cognition. And to measure the gut microbiome, we're going to be looking at bacterial diversity. So getting to know what kinds of bacteria are in the gut. And we're going to be doing this in mice. So here's our experimental design. We're going to get a random assignment of mice to experimental groups. Love to see random assignment. That's statistically very important. And we are going to subject these randomly assigned mice to 4 different experimental conditions here, right? The first one is going to be a normal diet. So this is just the stock diet that you could buy from these veterinary labs. Then normal diet plus forced exercise. So what happens is that we're going to pluck a mouse onto a running wheel, and we're going to automate the running wheel, so like a treadmill where the treadmill is automatically running, and that mouse is going to get an hour's worth of forced exercise. Yeah, an hour's worth of forced exercise. The third group is going to get a high-fat diet consisting of 60% lard, and then the fourth group is going to get a high-fat diet plus forced exercise. And then we are going to measure anxiety / cognition and measure microbiome populations after that. All right, so 4 different experimental groups, each with a different condition for diet and the presence of exercise. So the first thought-provoking question I have here, chat, is how do you feel about high-fat diet being 60% lard? How do you feel about that? How do you feel about the kind of data that we'll be able to get from this experiment? Couldn't forced exercise stress the mice? Ooh, Spepik, we're going to talk about that in just a little bit. But certainly that is something, when I read that in the methods, and this is why, biology students out there, if you're ignoring method sections, you're doing it wrong. Always, always, always read the method section for stuff like this, right? Because if you just take the author's word, they go, oh, we had the mice exercise, and if you don't critically think about what that means, you'll never catch stuff like this, right? So they're doing forced exercise as their paradigm. How much fat do mice normally eat? I think the normal diet is 10 to 20% fat, but I'm not super sure from memory. I think high fat should also be high protein, to be honest. Why do you believe so, Alice? What would that change about their question here? How does the microbiome react to high fat diet? We don't know as of this paper. This is part of what the paper is trying to investigate. Isn't lard pure fat, so we might only see the effect of more fat. That is true, but we're trying to see the effect of diet. Does this fit with that idea? Is this okay? Is this okay that we're doing 60% lard? I guess I should say my question should be, why did they use lard rather than other kinds of fat? That's great. That's a great question. I don't know why they chose lard. It might have been easy. The mice might have preferred to eat lard over something like vegetable oil. It's a good question. I don't believe they give an explanation as to why they chose lard, but why might that be important, or why might it not be important? It's kind of an interesting experimental design choice, and there are different ways to interpret this decision, and I want to sort of talk about the nuance behind experimental design. That's why I'm asking this question. Can you do 100% lard safely? Well, you would be missing a lot of, macro and micronutrients, because you still need protein. So protein still has to be a part of that diet. Calorically or by mass? Great question, Marlon. This is 60% lard by calorie, not by mass. So 60% of calories coming from lard. It's important because we're trying to enact ketosis, not quite, so we don't have a specific hypothesis about ketosis here. I was just wondering if anybody had particular ideas as to whether or not this is problematic here. Isn't protein essential for function of things like muscle? So overall, overly high fat but without protein, especially with exercise, could cause functioning issues, I guess. That is certainly true. But remember, we are thinking about changes to the microbiome and to behavior. So I don't really see a direct hypothesis there. Fats are large molecules that may not be able to be broken down by gut microbes. That is fair, but does that make this experiment problematic? It is saturated fat. Maybe they use it because of that, perhaps. Although they don't have a particular hypothesis between saturated versus unsaturated. Otto. Yes, that is the answer that I'm looking for. 60% lard sounds unrealistic for translation to an actual person changing their habits. Yes, that is exactly the answer. Thank you, Otto. So, high-fat diet being 60% lard. Critics of this paper could say, oh, this is bunk, because no human would have a diet that is 60% calorically from fat. And I know that nobody in chat other than Otto had said this, and that's not to blame Otto, but I think Otto kind of figured out what I was fishing for in terms of the nuance here. So when you see something like this in the wild, when you see scientific critique and they go, oh, this study's done in mice where they give the mice all sorts of unrealistic conditions, this can't be applied to humans. My answer to that is, okay, that is true. Studies done in mice under unrealistic conditions can't directly be applied to humans. Nobody's claiming that. But if we want to generate an effect, we kind of have to do certain extreme things, right? So while 60% lard is not a realistic experiment for a human, to get an experimental effect, we need to sort of pump up our experimental conditions, right? And you would never be able to get away with doing this in a human because you would violate certain laws of ethics. You cannot do harm to people when you do experiments, right? So in mice, this is the only way that we can find out the effect of a fatty diet in mice. And mice don't actually live that long. Mice have a lifespan of about 2 years. So if you wanted to do a more realistic simulation of what a high-fat diet would look like in a mouse, you might not see an effect because they might die before you see anything, right? So keep in mind that when you do experiments in mice, you have to think about the conclusions that you can draw in mice, and then you can think about the conclusions that you can draw in humans, right? So thank you, Otto, for allowing me to talk about this teaching little moment. Just like if you see angry Redditors online dismiss any mouse study because it's unrealistic, well, yes, but no. So just want to kind of get that out there. Do you have any thoughts on that, Myelin?
Well, I think I'm starting to understand some of the mindset that y'all have when you're going in with stats a bit better.
Yeah, because when you
Mhm.
study people, yeah, I mean, like, as psychologists, you have the ability to do controlled experiments, double blind, placebo, all these wonderful experimental tools at your disposal. But if you don't find an effect, that's kind of the end of the story. Whereas in
Mhm.
mice, because we have more control, because we have a shorter, maybe I shouldn't say shorter, but because the model organism of a mouse lives much shorter than a human, you kind of it's more like we will do things that are too much for a human so that we can generate an effect, and then if the effect exists, we can then try to do it under more realistic conditions. But the
Mhm.
first step when you want to find an effect, the first step is to be a little extreme. I talked about this in the vaping paper that I did way back in my channel's early history, one of the first journal clubs I've ever done, where the mice were exposed to vape, like e-cigarette vapor, for like 6 hours a day, and angry Redditors were like, oh, these mice, they were exposed for 6 hours, no one's sitting in a vape chamber for 6 hours, can't draw any conclusions for humans. Well, yeah, but again, mice don't live as long as humans. If you want to see an effect, you kind of have to be extreme, and if there is an effect, then we can invest more money to actually figure out if it is applicable to humans. So just wanted to get that out there.
Mhm. I mean, you also have me thinking about why you would want such a big effect size outside of that. It's because y'all, well, I think we'll probably talk more about this later, but it's difficult to get a large sample size. And there's a lot of things that you can do that will affect your likelihood of finding results, even with a small sample size. And trying to make as big of an impact as possible is absolutely one of them.
Yeah, it's expensive, and for animal welfare purposes, you don't want a high end, right? If you've seen an experiment and they're
Yeah.
doing like 50 mice, that's so many wasted animal lives. Like, despite, okay, we're feeding these mice 60% fat, you could argue that that's not cool. But presumably, if you're on my channel, you kind of get that animal experimentation is a necessary part of the biology research pipeline. Minimizing the number of animals that you use is really important for biology because, believe it or not, we don't want to test on animals. There's just no
Mhm.
better alternative if you want to ask questions about humans, right? You're not going to ask 20 random people to adapt a 60% fat diet so that we can study the gut, right? Like that's not practical or ethical. Society has kind of determined that this is an okay line to draw.
Hmm. Hmm. I'm
Okay.
also thinking about how you said most of my work is correlational, and that's true because, like you said, it's not ethical to do a lot of true experiments for a lot of questions in psychology.
Exactly. Yeah. Yeah, for sure.
Hmm.
Okay. So, yeah, we got to start in mice, and sometimes starting in mice means doing things to mice that you wouldn't normally do to a human. So, let's look at the data. Okay. So, in blue triangles, we have the normal diet, ND. In green, we have normal diet plus exercise. In red, we have high fat diet. In orange, we have high fat diet plus exercise. So, the researchers measured the weight of these mice over time, for these 4 groups over the course of 24 weeks. So not 24 weeks, but starting from the age of 8 weeks to the age of 24 weeks. So this is basically mouse adulthood, pretty much. 8 weeks. Yeah, 8 weeks they should be sexually mature. So this is like an adult mouse over the course of 24 weeks. Yeah, yeah, yeah. And so they were tracking their weight over time, and so this is the data that they got. Why would the researchers be interested in measuring the weight of these mice? What is the experimental goal here, chat? What happened in week 15? I don't actually know why there's a gap here.
Oh, that is weird.
Good eye. Good eye. A measure of metabolic storage. There's a simpler way to say that, maybe. Wibbles to see if they gained weight. Definitely true to see if they gained weight, which implies what? Does weight affect behavior? Not quite. We're not there yet. To see if there were trends. Maybe the weight didn't increase uniformly. That's overthinking. I'm more on the side of Wibbles right now. If they can metabolize the diet effectively, that's closer. Yeah, Wibbles, I think, is the most correct here. It's okay. So the reason why I think Wibbles has the most correct answer here is they just want to make sure that their high fat diet works. Like to confirm expected effects, exactly, Spepek. They just want to confirm that the mice behave in basic ways that are already proven by the literature, right? Mice with a normal diet, you should see a normal increase in weight as the mice age. It should probably plateau in their adult weight. 8 weeks, it's a young mouse. They can still grow a little after that. So this is somewhat normal. And then no diet plus exercise. Well, it makes sense that if you force the mouse to run for an hour a day, that those mice would be a little more lean than the mice that were just eating a normal diet. It's actually really funny. If you dig into the methods section, the sedentary mice, the blue and the red mice here, in order to control for other experimental factors, they still put them on the running wheel, but they actually put them on the running wheel and then turned on the running wheel to run so slowly that they couldn't sleep. So they weren't running, but the wheel was slowly automatically turning just to keep them awake. And that was their sedentary condition, which I thought was really funny. Yeah, so they're basically confirming that their high fat diet causes the mice to gain weight, which means that everything is working as expected, and that exercise decreases it. So that's good that they did that, just to make sure that the diets were behaving as they are expected to. So now let's take a look at whether or not these mice had differences in their anxiety. So when you measure anxiety in a mouse, you could do it in a couple of different ways. The way that this paper decides to measure anxiety is with the light-dark interaction test. So what this is you have a box, and this box is connected with a little hole in between, and there is this large portion of the box that is lit up uniformly, and then there is a portion of the box that is completely dark. And the measure of an anxious mouse is going to be a mouse that spends most of its time in the dark box and less of its time in the light box. The thing is that mice are naturally very curious creatures. Of course, mice evolved to be curious because being curious can help you, acquire resources. Again, from an evolutionary theoretical perspective, I don't want to make it seem like I know that for a fact, but just like from evolutionary theory, you could believe that mice that exhibit curiosity are more able to acquire resources. But an anxious mouse is maybe going to prioritize their own safety and spend more time in the dark room. They're just like me, for real. So that's how the light-dark interaction test measures anxiety. So how much time does the mouse spend in the dark room versus the light room? And when they're in the light room, how often do they move around in the light room? And here's what they found. So in blue, we have the no-fat diet, and 30% of the time that they were in this test, they were in the light room. And in the high-fat diet, they'd actually spend a little less than 20% of their time in the light room. So we can conclude that the high-fat diet causes mice to spend less time in the light, which could be a measure of anxiety. Exercise does not majorly impact anxiety for mice consuming a normal diet. So if we compare blue with green, we can actually see that the exercised mice spend just as much time in the light room as the mice that were not exercising. And exercise does not rescue the increased anxiety that the high-fat diet mice experienced. So this is already a very interesting result. Myelin, when you read this, were you at all surprised, or were you like not, or did you not like have a bias going in here?
I don't think I had much of a bias coming into this, so I wasn't terribly surprised by the result, I would say. Mainly because I didn't really have any expectations.
That's fair. I also didn't really have expectations. It was only after finishing the paper was I like, hey, that's kind of interesting.
Mm-hmm.
The exercise is so extreme, sleep deprivation could have biased the results. It's not really sleep deprivation because they were only running for an hour, right? The mice still have the rest of the 23 hours to get a full night of sleep. It's just for that 1 hour they were exercising, and then they returned to their cage. It might just be that they don't want to move as much. They chunky. So Spepik, you touched upon something that I'm going to get into again in just a little bit. I'm loving this because we're thinking like scientists, like angry reviewers. Exactly. They could just have gained a lot of weight, and maybe they're not very mobile anymore, and maybe they just don't want to leave the dark room, right? Like that's very possible. We're going to see how the authors address that in just a little bit. Wouldn't the high-fat diet make them less mobile? Yeah, exactly. So exactly what Spepik is saying. You would think that, right? Like that is a reasonable and smart criticism to have of this experiment. All right. So here's another metric. So this is light zone distance. So this is how much they traveled within the light zone. And it roughly mirrors the percent time in light that the other anxiety test shows, right? So they lose a statistical significant comparison here, but overall the trends look very similar to the other experiment, right? Are they depressed? Mice are social creatures. Yes. Oh, another fantastic hypothesis, right? Very, 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 very smart questions that we're asking in the chat here. So much so that I don't even have to like probe you to think this way anymore, right? Because I see the repeat names. You're all already naturally poking holes in the research, and I love that. So the authors will address some of your questions, and I believe that's. Yeah, see, I was going to prompt this, but Chad is already getting ahead of me. So what's an alternate hypothesis for high-fat diet mice spending less time in the light zone? Plenty of great hypotheses in the chat. So yeah, maybe high-fat diet mice have low energy, you know? Certainly if I just had a pizza for dinner and I feel very groggy, like maybe they're lethargic or sickly, or maybe they're just not curious, right? Like maybe this isn't a measure of anxiety. Maybe they just don't care about the light room, right? All of these things could be true. So what do the authors do to rule those out? Because science is not about proving your hypothesis right. So this is something that it took me a while to wrap my own head around. You cannot prove things to be true. You can only prove everything else to be false. Would you say that's accurate, Myelin?
I wouldn't even say you can prove that other things are false. I would say we can't prove anything's true, but we can provide an increasing amount of evidence for something.
Yeah, that's even more conservative than what I would say. I would definitely say, like, from my perspective, the approach... Okay, let me back up. The approach that I think that scientists should take is not to verify hypotheses, but to falsify them. And if you cannot falsify hypotheses, then it has stood the test of time. And that I've mentioned this before, is that good science, you'll know it when you read it. Good science tries to break hypotheses. And these controls are an example of the authors making an attempt to break their hypothesis. So they could just do these experiments. They're more anxious, and they could have just moved on, right? They could have just moved on. We see what they're like. They're anxious. I'm not going to think about it too much and move on. But of course, if you're a good scientist, then you will try to break your own hypothesis just by asking the same questions that you have asked in chat. So here's what they did. They did something called the open field assay, and it showed that high-fat diet move around normally and show normal levels of curiosity. So in panel A, they took each of these mice and just plopped them in an empty cage and just let them go and observed how much they moved. So you can see that when they're just allowed to explore a novel cage, that all of them actually do roughly explore the same amount, same distance. So they're sniffing around, again, mice are very olfactory motivated animals, so they do a lot of smelling of their environment. So a mouse that's introduced into a safe new environment is going to spend a lot of time exploring. And so it looks like it doesn't matter what kind of diet you get. It also looks like it doesn't matter what amount of exercise you get. All of these mice appear to move around in this environment the same. So that rules out the lack of curiosity, right? Because they're exploring their territory, and also rules out gross motor dysfunction, right? Because they're all moving the same amount. So this is a really elegant way, I think, to rule out two of those alternate explanations.
Mm-hmm.
Assay, that's just another word for test. Okay, so this panel B, this is rearing. So when a mouse is sniffing around in the environment, it can sniff at horizontal level, but also it wants to get like height information as well. So you can see mice when they're exploring, they'll stand up on their two little legs and sniff the sky, right? Lots of vertical exploration as well. And in those cages, there are photo beams, like little lasers, and you can actually detect, like in spy movies, right? When you're trying to like swap the gem or whatever, those same photo beams, you can detect if those beams have been broken. And so the more photo beams are broken, the more that mouse is standing on the rear end sniffing the sky. And it looks like overall, it looks like all of these mice are rearing at the same rate. So this is a more direct measure of curiosity. Although you could say that the high-fat diet plus exercise group is trending towards less curious, but it's not statistically significant, so we move on. Or at least that's how I treat it. I don't know, Myelin, if that like strikes you as not okay to do.
I mean, normally I would say that we probably shouldn't interpret, like we should just straight up just say we have no evidence that these are different, and I would get upset at the word trending, or maybe upset isn't the right word, but bulk at the word trending. But in this case, because their sample size is so low, I would say it's, yeah, we have no idea. It could be they just don't, there is a difference there and they don't have enough power to detect it.
Yeah, that's why I like how you phrase that. It's like they're allowed to say that there's no difference because their testing shows that there's no difference. But like, if you're being critical, you look at this and you might go, I wonder what would happen if you did a couple more experiments.
Mm-hmm.
Why might high fat plus exercise affect rearing if it is? I don't know. We're going to find out what the authors suggest later on in this paper. All right. So that should, and I guess we can pause here. And just say, does that sort of address your concerns, chat? Or are you not satisfied with the description of these tests? Sorry, but I really need to sleep. Have a good sleep. Sleep is very important for the brain and very important for your cognition. Have a good rest. But yeah, chat. So hopefully these two experiments have addressed some of your hypotheses. And I love this when I read this in papers. Like I go into a paper and I don't want to be mean, right? I don't want to be mean. And so, but you have to as a scientist, right? Like when you read a paper, your natural inclination is to be careful when you interpret data, right? And you want to be critical because that's what science is. But I don't want to be mean. So when I say, oh, I wonder if it's because they're less healthy, that they don't move around as much. And then when the authors do that experiment, I'm like, yes, controls. We love controls.
I had the same reaction to this paper. I was worried about social effects because that's the kind of psychologist I am. And then they measured that. They measured sociability. And I was like, oh, that's great.
Yeah. So,
cool ideal.
Oh, do I not have that here? I could have sworn I had the sociability slide in here.
I think you did mention it before. I don't know.
I mentioned it, but I don't know if I have that figure. That's kind of a bummer. I don't think it's in the slide. I thought I had it in there. I'm so sorry. They also measured their interactions with novel mice, how they explore other mice, and I believe their conclusion was similar, that they're overall as sociable as their counterparts. So all four groups of mice were roughly as social as the other groups of mice. All right. So they measured anxiety, and it looks like it might be a true measure of anxiety because it's not a measure of gross motor defects or curiosity, vaguely, right? So what does that leave us with? Probably something to do with anxiety. So we'll say that exercise, or sorry, I'm already forgetting the effect here. High-fat diet, right? So we can say that high-fat diet probably affects anxiety here, unless this can be explained through some other mechanism, but I can't really come up with any. Okay, so we tested for anxiety. Before we move on, Myelin, is there anything you wanted to add up until this point?
Um, Not that wouldn't knock us horribly off track. So.
We might. I mean, is it like the stats stuff that we can get
Yes.
into? Okay, we can get into that later.
Yeah,
Okay, great.
we'll save that.
Okay, so high-fat diet causes mice to look more anxious. I'll say that, to look more anxious, to appear to be more anxious. What about memory? So here's how the experimenter set this up. So they wanted to test a specific kind of memory called contextual fear memory. And this was induced with an experiment called the foot shock experiment. So here's how this is set up. So you plop a mice down in a cage and you play a white noise tone. So this is our conditioned stimulus. So I can have our psychologist explain what these mean, maybe at the end of this graph. And then they wait 28 seconds. So they play this white noise, 28 seconds pass, and then they electrocute the floor and give the mice a little bit of a foot shock. So nothing to really hurt them, but certainly enough for them to get startled, right? So the foot shock is the foot shock is our our unconditioned stimulus. I think I got that right, right?
Yeah, okay. Yeah. It's been a while since I looked at conditioning, but that sounds completely correct.
Okay, great. I guess I shouldn't have just assumed that you also did behavior. That was maybe a little science racist of me.
I mean, the type of behavior that I do is not the conditioning type. It's very different. So.
Gotcha. Yeah, that was me making overgeneralizations, which is maybe poor form.
Nah, you're good. I mean, you're not assuming that psychology is entirely mental health, so you are already leaps and bounds above.
Yeah, that would do it. All right, yeah. So Pavlov, right? So this is like Pavlov. Is it like Pavlov? I don't actually know.
I would say it's fairly similar to Pavlov. This is what's bugging me. There's some other person who did stuff with conditioning, and I can't remember if this is Pavlov or the other guy.
There's classical conditioning, and then there's operant conditioning, right?
Yes.
Where there's a punishment or a reward.
Yeah, okay, yes, this is absolutely Pavlov. Yeah, Skinner was the other person, the operant conditioning. Pavlov was this one.
Gotcha. So sorry, is this Pavlov or Skinner? Now I don't remember.
Pavlov.
Okay.
Skinner had his Skinner box where he wanted to raise children in a box.
Oh, the fun history of psychology.
We had a lot of fun before ethics. That was sarcasm.
Tone sarcasm. All right, so the idea here is that a mouse with better contextual fear memory is going to remember that the white noise means it's about to get shocked in the foot, right? So that's kind of what they're testing here. All right, so you play this white noise, you get the mouse to learn that white noise means foot shock. And then when you play the white noise, you should see a fear response, which in mice is freezing. So if the mouse has really good contextual fear memory, they're going to freeze in response to that white noise. And how much they freeze is an indication, a correlate of their contextual fear memory. So how does high-fat diet and exercise affect that? So percent freezing, again, is our metric of contextual fear memory. The longer they freeze, the more the quote unquote, the better memory they have, quote unquote. Okay. Being very careful here. So if we compare blue and red, we actually see that high-fat diet doesn't appear to have any effects on percent freezing. So overall, it looks like being on that 60% lard diet doesn't affect how well these mice respond to the conditioned stimulus, the white noise, in preparation for that foot shock. But what did appear to change is between the normal diet and normal diet plus exercise, the exercised mice that were eating the normal diet had a longer freezing time. They were more, I should say, maybe I'll say it this way, they were more affected by the conditioning. Whether or not that means they have better memory, I actually don't know. I actually don't like the foot shock as a metric of memory at all. I would have preferred like a water maze of some kind, although that might be kind of hard. Actually, would water maze be better? Ooh, maybe they did foot shock for a very good reason. I'm just kind of figuring this out live. So what's the problem, chat, if they were to do a maze instead? What's the problem with doing a maze in this particular experiment? I'll give chat a couple seconds to think. Because I don't like the foot shock because, you know, what if they just don't care about getting shocked? Like, that's a perfectly valid thing to happen. Like, they don't, or maybe what if their pain response is dulled, or maybe their hearing is bad, you know, all these other things that could be correlates. And I would have preferred a maze instead to figure out their memory. But why might a maze actually not be very good here?
I'm sorry, I'm reading chat and I'm liking some of these answers.
Yeah. The weight of the mice would influence the result. Yeah, like if you're big chungus walking down a maze, it'll just take you longer to find what you need to find, right? And typically with mice, you would do a water maze, and so like if you're big chungus, you might not be able to like swim as well. Sure, they might move around voluntarily as often as their peers, but in terms of a situation where movement is timed, the heavier mice are probably going to have a harder time. So maybe they had to do foot shock. That's kind of interesting.
I'm sure they could have. There's got to be something other than foot shock. Like, I mean, I'm not as familiar with mice methods, obviously, but
there's another oh, okay, so there's another experiment that you can do. I'm remembering this from like grad school. So I think it's novel object recognition. So
yes.
I think you can put a mouse in a cage with an item that they're familiar with and an item that they're not familiar with, and you can measure how often they go to inspect the unfamiliar object versus hang out with their familiar object. That might be a little better. I don't know why they chose foot shock.
Yeah, I mean, I'm interested. I like the novel object one because the foot shock, they're interested in memory and anxiety. And I feel like foot shock, the idea is that they'd freeze more as a fear response or as an anxiety response. So it feels confounded with their measures of anxiety. I can't tell if any effects due to it are due to memory or anxiety or both.
Yeah, that's really true. Yeah, what Myelin said was great right there. Like contextual fear memory, like fear and anxiety go hand in hand. And if we think too hard about it, we're going to end up with an issue where we have these confounds of like, are the high-fat diet mice just more anxious? Because we already know the high-fat diet affects anxiety. So is this really memory or is this just general anxiety? It's really hard to tell. I would have preferred that they do novel object recognition instead. So that is, I think, maybe my personal biggest gripe with this paper is I just don't like foot shock as a measure of memory.
Mm-hmm.
It would have been interesting to test more types of memory. Yeah, because with novel object recognition, I don't think that has anything to do with how heavy the mouse is. We already know that they move around normally, so you can't. So that's probably not going to be an issue. Yeah, I just don't like the foot shock thing. And you bring up a good point about confounds. All right. So here are the conclusions so far. We're looking at behavior first. So we've got a line from exercise to brain and diet to brain. And so we learned in the data that high-fat diet increases anxiety or increases the appearance of anxiety in mice compared to a normal diet. And exercise does not appear to have a major effect on anxiety in either diet condition, right? So it looks like anxiety is having to do more with your diet than with exercise, not your diet, but in the mice's diet than exercise does. So exercise might improve contextual fear learning. Sure, but you know, with the asterisks that Myelin and I have brought up, in mice fed a normal diet. So it looks like comparing a normal diet to normal diet plus exercise, that exercise improves the acquisition of the conditioning of fear memory. How do you measure a feeling of unease? Light dark test. That's pretty good for mice that don't want to. That mice that want to be relieved of their anxiety will choose the dark space. Okay, so just to fill out our little diagram, exercise kind of improves memory and high-fat diet increases anxiety. I think I'm okay with that. I think that might be the strongest conclusion so far, in my opinion.
Mm-hmm. Mm-hmm.
All right. We have another axis to look at, and that's the microbiome, right? So how does exercise and diet affect the microbiome? And so this is where we get a little a little bit of like the grifter-esque aspect of this. Like not blaming the scientists, right? But like this is where grifters can really latch onto. In fact, if you look up the social media sites that have shared this article, a lot of them are advocating for health foods. And there's nothing wrong with people trying to sell you health food. There's nothing wrong with it. But when they cite this paper, it's like, I don't think you're being totally honest. But in any case, let's look at what they found about the microbiome. Is a stronger fear conditioning a good or bad thing? Like if someone learns to fear something faster or slower, is that helpful or harmful? I don't think the authors make a hypothesis in this paper, and that's a very complicated question to answer.
Well, I think either extreme is definitely maladaptive.
Yeah, I like that word, maladaptive. You want an adaptive anxiety response, not a maladaptive anxiety response. So if it makes your life harder, then it's probably bad. If it makes your life better, it's probably good.
Yeah.
Can you keep track of populations of various microbes in the intestine? We are just about to get into how they did that, which is super cool. Okay, the other thing is that these mice were forced to exercise. So that's also interesting. So even through the exercise, the forced exercise, like mice being forced to run that wheel, it doesn't appear to have increased anxiety. So that's interesting to me. So maybe the data is even underestimating the effect. So maybe if we did a voluntary wheel versus no wheel at all, maybe we would see differences. I know that if you made me run on a treadmill for an hour, I would really feel quite stressed, but I don't know if my overall anxiety would change. My anxiety would probably change if I knew that I had to run an hour every day.
Yeah. I mean, and the relationship between anxiety and stress, they are technically distinct, but they are pretty highly related.
I love those correlations. Love those correlations.
Yeah.
Yeah. So, it's possible that there is an effect of exercise, but because they did forced exercise, maybe that kind of obscured it or something.
Yeah, and it could be that they were just making all the mice anxious in general. It would have been nice to see a baseline of like elective exercise, or like you said, like in general.
Yeah, so they do cite some papers that do a similar setup with voluntary exercises. I forget exactly what they said about those conclusions. But importantly, forced exercise means that you can give a very precise amount of exercise per mouse, which is an important experimental variable to control. So again, science, when you get to like very high level like this, it's like art in a way, right? Like we all want to think that, oh, science is like, you can't argue with science, science is correct, etc., etc. But the approach that scientists take is actually quite artful, right? Like each scientist has to be aware of the story that they are trying to tell and to craft their experiments based on the appropriate story. Not to say that they have bias or something, right? But like the authors wanted to see the effect of moving, not engaging in leisurely exercise, right? Just the idea of moving. And so you gain something by making it so that you can control that experimentally, but you lose some of that face validity because normal mice aren't forced to exercise, right?
I mean, at the end of the day, every experimental design is a balancing act. You are trying to decide whether you care more about internal validity, controls for confounds, or whether you care more about external validity. So does this actually reflect real life? And there's, like you said, there's no right answer. You're trying to find what you think is best for the question you're trying to ask.
And I think that's part of what makes science so fun. Like these experimental decisions, these designs, the ability to craft your own scientific story and to make those decisions, I think is part of what makes doing experimental work so exciting. Because it's your job to convince people that this is the correct way to do it, or this is the best way to do it in your story. And so while you have to acknowledge that there are trade-offs between forced and non-forced exercise, I don't find that to be very offensive, right? Like this doesn't mean that this paper is bad in my eyes. It's a choice and we should be aware of it. All right, so how do you keep track of different microbial populations in the gut? So gut microbiome sequencing has advanced quite a bit in the last decade. The way that it was done a decade ago is through fecal transplant. And so there are other methods now, but this is still used today, and it's pretty good, but there are some caveats here. So what do I mean by fecal sequencing of 16S ribosomal RNA? Okay, so we have a couple of different techniques: fecal sample collection, biopsy, luminal brush. So where's my slide? Okay, yeah, let's first go here. I'll do these slides in a bit of a different order. So the ribosome is, you could call it an organelle, I guess. Maybe not. No, you shouldn't call it an organelle. That's wrong. The ribosome is a protein RNA complex that reads mRNA, messenger RNA, and creates protein, right? So it is made of parts that are protein, like out of amino acids and stuff, and the other part of the ribosome is made of RNA. So you have ribosomes, bacteria have ribosomes, every organism that we know of has ribosomes. You can't have life without some machine making your proteins, and that's what the ribosome is, right? And so in order to test for genetic diversity of the gut, you actually sequence the RNA portion of this ribosome. So in prokaryotes, this is 16S RNA. So 16S refers to the size of the fraction that they isolated this in. It's just a bit of historical context. But each bacteria, their genetic fingerprint, you could call it, is through the 16S RNA, because every bacteria has a slightly different version of this ribosome. So you can collect a fecal sample. So, you just wait for the mice to poop. You take those little droppings and then you solubilize them in some solvent. Extract all the RNA that you can, sequence all of the RNA, use fancy computational biology to put those sequences together, and you have a massive readout of all of the different bacterial populations in that sample, at least the ones that have high abundance. All right. Welcome, Clockwork. Welcome. We're talking about 16S RNA sequencing. Do the ribosomes of different organisms interact with tRNA differently and construct proteins in different forms? So that's a great question, and is getting into some of the classes I took as a graduate student. Oh, tRNA is such like a complex topic, and I'm honestly not prepared to talk at length about how tRNA works. So transfer RNA, that's part of the protein assembly machinery, and I'm not ready to talk about that. Okay, so yeah, we take those little poops and we digest them, not digest them, we solubilize them, extract that RNA, and then we can get a genetic fingerprint of all of the microbes that live in that fecal sample. All right, so that is fecal sample collection. Nowadays, we can also do biopsy, so you can take a little biological sample directly out of the intestine, just like carve a little piece out, and luminal brush, so you can stick something up into the mouse's intestines and swab an area if needed. So there are different methods, and just like the foot shock and just like the forced exercise, there are choices to be made here, right? I personally don't love fecal sample as a way to measure microbiome changes. I'm not a microbiome person, but to me, this is like, I feel like we could do better. So why might I not love fecal collection as a way to measure the microbiome? Is fecal sample measurement an accurate snapshot of the microbiome in the mouse? Do some bacteria localize to different regions of the intestine? Yeah, that could be part of it, right? So the bacteria that exists in the small intestine and the large intestine are different, and the stuff that is going to be represented in the poop is going to be stuff that came out more recently, right? Yeah, the colon is a very different environment compared to the other regions. Not all the bacteria might end up in the poop, exactly, right? Like fecal sampling of the microbiome is fine, but there's a lot of like nuance that doesn't get captured in that sort of sample. Does fecal sample oversample / undersample those in the colon / large intestine? Exactly. Like you might not get a totally accurate picture of what's going on in a large portion of the actual gut. So when it comes to measuring the microbiome, I would prefer there to be a little bit more of a direct measurement like a biopsy or a luminal brush. But why might that also not be great for this particular experiment? So biopsy, cutting out a little sample of, whatever region of the gut that you're interested in, or a luminal brush, sticking something up the mouse's, and then swabbing something from there. Like, why would that not be great either here? Biopsy is invasive. Yeah, biopsy is invasive, right? Like, you don't want to hurt the mouse unless you really have to, right? That's just baseline biological ethics. Like, don't do things that hurt animals if you don't have to, and picking up poop is not going to disturb the mouse at all. But B, as icosahedron and Alice have pointed out, biopsies is too invasive, right? If we're talking about effects on anxiety and effects on the microbiome, right from the moment you administer whatever anti-inflammatories that you need to prevent infection in a mouse when you're taking a biopsy, if you have to at all, or as you're taking a biopsy, you're already invading in the immunological niche of the gut to begin with. You're affecting the microbiome in real time when you're collecting a biopsy. So, none of these are really great. If I had to pick one, I actually would pick fecal sample, just because I would rather have a good understanding of the poop microbiome versus a messed up sample of the actual gut. Does that kind of make sense, chat? Hurting the mouse would affect later behavior. You might not have to test the same mice that get the biopsy and the same let me put it this way: the mice that get tested for anxiety don't have to be the same mice that get biopsied. In the same way that your hormonal / transcriptome vary widely throughout the day, taking a single sample of a single region of something as broad as your microbiome is inherently limiting. That's also true, right? If you're not taking multiple samples, then you're only getting a very brief screenshot, and that might not be very informative either. But yeah, so I don't love fecal sample as a way to measure the microbiome, but it's not the worst. And certainly for this thing about like anxiety and like that kind of thing, especially because fat, we know, is an immunological signaler, right? Fat contributes to the immune system or affects the immune system in interesting ways, and if we're hurting the immune system, we might be screwing up some of the effects that fat might be having, right? Like, this is I think a good choice, even though it was a choice that I initially reacted quite negatively to. This is such an interesting discussion. Yeah, I love science, man. like this is something that I get on the soapbox every stream, but like the big educational giants that make all the educational content you love, they don't really get to talk about the nitty gritty, and professional scientists love this kind of nitty gritty. And it's such a shame that we don't get more opportunities to share these nitty gritty things with people who aren't trained in this kind of stuff. How does fat affect the immune system? From my memory, fat can be generally pro-inflammatory, so activating, although I don't want to put too much stock behind that because that's not my primary field of research. Latest render just finished making. Ooh, make sure that you post a link in the Discord when you have that video up. Very cool. Okay, so any thoughts on sampling the microbiome? So you just heard Myelin, me discuss, and we didn't talk about this beforehand, but like, does this kind of like make sense, this idea of like every technique has its own biases? And I'm curious to hear what you think about that.
Absolutely. I mean, it's very consistent with a lot of the psych techniques that we have, and I thought it was very, very fun learning about these techniques. And just know, whenever you ask Chad a question, I'm trying to come up with answers too.
That's awesome. Marlon, are there other ways of imaging through the structures of the body? So bacteria are really small, like really, really small. And the sort of like gross anatomical imaging that you can do in a live mouse, not exactly appropriate for structures that small, at least to my knowledge. Okay. Yeah, so I don't love it because you can't distinguish where the bacteria come from. They all get looped in. But yeah, biopsy, endoscopy, brushing, they're invasive and stressful. And if stress is having an effect on the microbiome, then that's like, it would be good not to capture that in the data collection. Okay, so we collected the sample and now we're analyzing it. So again, we talked about sequencing the 16S RNA. So getting whatever sequence the RNA is from these bacteria, that's how we determine what is what. And so here are their data, right? Bacterial changes in the microbiome due to exercise and diet are hard to characterize. And I, behind the scenes, agonized over this particular, not this particular figure, but a figure that's coming up, because this to me is very hard to make useful in my brain, right? So let's take a look at panel A. So this is looking at a specific type of bacteria, the Streptococcus family. And it looks like in a no-fat diet, there's like non-detectable amounts of Streptococcus. So a relative abundance of 0.000. 0.0000, right? So you basically don't detect Streptococcus, then you feed them the high-fat diet, and bang, the Streptococcus level goes way up, going from non-detectable to quite high, maybe? Not a microbiome person, so I don't know if relative abundance of 0.0006 is like a big deal, but the authors do refer to this as a bloom of Streptococcus. So that's pretty interesting. And then in B, we get some different effects, right? Like no fat diet and high fat diet. You get an increase in firmicutes, I don't know how to say any of these bacteria names. Firmicutes sounds kind of fun, so I'll just call it that. And then between the two exercised groups, you also see that difference. So that's like kind of hard to understand. So it looks like high fat diet in general might increase the population of firmicutes. I'm going to butcher all of these bacterial names. It looks like high-fat diet plus exercise decreases bacteriodetes. How do you say any of these? I should have looked these up beforehand.
Clearly just let me pronounce them. I'll just do it so hilariously wrong, but I have an excuse.
Yeah, you have an excuse, and it would protect my brand image. That's good thinking. And then in D, it looks like, high-fat diet completely abolishes tourniquet population, and looks like maybe exercise restores some of it a little bit, although they don't actually make that comparison, so who knows.
I mean, that comparison's there, right?
Between sorry, you're right. Yeah, it is there. So this is a really hard graph to make any sense of, right? You have some bacterial populations going up in certain groups, some going down. I don't really see any super obvious trends. I don't know, Myelin, if you like noticed anything worth talking about here. But when I looked at this figure, I was just like, okay, high fat diet, Streptococcus, cool. I don't really know what to say other than that.
Yeah, I don't know. I mean, whenever I saw bacteria names, I admit my brain glossed over a little bit, but I think one of the interesting things is something that Marlon pointed out. Not just Marlon, who else was talking about this with the... Oh gosh, I'm scared to say Streptococcus because I'm going to mispronounce.
No, that was good.
Oh my gosh, all right, awesome. But yeah, it's so interesting that and again, like you said, I have no idea what scale these y-axes are on, and p-values are not effect sizes. And based on the y-axis in A, it looks like it's probably exaggerating how big the effect is relative to the others, given the numbers we're seeing there. But it is really interesting that we're seeing this significantly higher amount of in the high-fat diet condition, but when they exercise, we are not seeing that same thing, which is interesting. It's not nearly as big of a difference with the normal diet, but that's weird.
It is really weird, and there's like a good reason, I think, that all of this data is so like hard to interpret, and I think this is the figure that like I was just like agonizing, just like messaging Myelin, going like, I don't like get this. My brain doesn't like understand this at all. So yeah, this MDS clustering analysis, I was just like looking at it, I was like, I kind of get what they're going for, but I need to be able to explain this to my audience. So MDS is a type of dimensional analysis. So the microbiome is really complex, right? So you expose the microbiome to these 4 different conditions of diet and exercise, and the researchers want to find out if these effects cluster in any kind of way to determine what effect diet has and what effect exercise has, to be able to sort of untangle the effects in a kind of way. So in this clustering, you have these dimensions, and I don't believe they define these dimensions. It's okay. It's not really necessary that we know what these dimensions are. But the idea here is that these mice, their microbiomes cluster in 4 distinct groups, which implies that diet and exercise appear to affect that microbiome in unrelated ways. So this is, they just said that, and when I first read the paper, I was like, yeah, sure, that makes sense. And then I really read it, and I really thought about it, and I was like, I actually don't know why that is. So by clustering in 4 different groups, it implies that diet and exercise have different effects on the microbiome. And so if they have different effects on the microbiome, it would make a lot of sense that this data doesn't appear to have like really consistent trends amongst diet and exercise. It's because diet and exercise are doing different things. And so I'm going to try to break it down why this data clustering into 4 different groups means that diet and exercise are acting in different ways. So principal component analysis hurts my brain every time. I have to reread how it works every single time. I never understand it for more than like half an hour, and then it just vanishes from my brain. But this is, I think, what the authors are trying to get across. Let's assume that diet has no effect on the data, and only exercise has an effect on the data. So if diet doesn't matter, but exercise does matter, then we can assume that the data will cluster into 2 unique groups, right? So in this chart, diet doesn't matter. If you exercise, you get group 1 effect. If you don't exercise, you get group 1 effect. If you do exercise, you get group 2 effect, right? So we can shorthand this by saying maybe group 1 is like bad microbiome. Shorthanding it. It's way more complicated than that, but let's just simplify it that way. And group 2 is good microbiome.
Oh no.
Okay.
Sorry.
PCA decomposes data into vectors, which are then the principal components. Yes, that is my understanding of PCA, but I don't really practically know what that means. I have to teach myself, I have to walk through what that means every single time. So let me explain it like the silly biologist that I am, right? Ignoring vectors and all those things. Let's just think about it in terms of unique groups, right? So diet doesn't matter, but exercise does. You get 2 unique groups. Let's take case number 2, where diet and exercise have the same effect, so additive effects. So if diet and exercise affects the microbiome in the exact same way, we should actually expect to see 3 different groups, right? So no exercise and a normal diet is group 1. High-fat diet and exercise is also group 1. So if you have one and not the other, then you're the same, right? So along this diagonal, you have group 1. No exercise and high-fat diet. You can call that like the worst microbiome, maybe, group 3. And then normal diet plus exercise, this is like the healthiest you can be. You should see 3 distinct clusters of population, right? So that's case 2. But we don't see 3 or 2 groups, right? We see 4 different clusters. So what does 4 different clusters mean? 4 different clusters. Now this is getting very complicated. Now we can no longer shorthand it with healthy microbiome, unhealthy microbiome. Now we just have to go with colors. Because it's too complicated to come up with 4 groups, right? So if diet and exercise are doing different things to the bacteria, then each individual intersection of these columns should have a different clustering. So no exercise, normal diet, that should have its own unique effect. Normal diet, exercise, own unique effect, group 2. No exercise, high-fat diet, own unique effect, group 3. Normal diet, high-fat diet and exercise, own unique effect, group 4. So that is why when they clustered this data, that they said that because they cluster as 4 non-overlapping groups, that diet and exercise have orthogonal effects. That was back in the title, orthogonal, meaning perpendicular. Meaning that they're not like, they're separate. They separate from each other. They diverge. right? So instead of clustering in 2 or 3, we get 4 unique groups, which implies that they are working the microbiome in unique ways. I wish we got a baseline for a normal mouse microbiome. So that would be like normal diet, voluntary exercise is the closest that I would think a normal mouse microbiome would be. Notionally, that would be normal diet. Yeah. Like in their experiment, the closest would be normal diet, no exercise, but a better would be normal diet, voluntary exercise. Aren't orange and red linked together as they are very close on the graph? Yeah, Myelin and I, we kind of talked about that a little bit. I don't know what your thoughts are, Myelin. My thought is they did the math, they did a monster math. I'm not going to argue with them.
The reporting isn't enough for me to really say. I mean, you can look at the error bars there, and the error bars are not overlapping, and they have asterisks, which usually indicates significance. But it's not clear what the asterisks are referring to, and it's not clear if the error bars are like standard errors or if they are 95% confidence. So it's, yeah, I can't tell. It could be that they're statistically distinct. It could be that they're not, particularly the last points for week 24 for the high-fat diet and high-fat diet with exercise.
I think
those
maybe
look like overlapping error bars.
So error bars in biology are usually standard error, but I don't know if they mentioned that in the paper. I usually assume they're standard error.
Okay, that makes sense. Yeah, so standard error is the estimate of how far off you are from the true population value. So it's kind of a measure of sampling from the true population.
But I think we are talking about the clustering analysis in terms of the
the clustering analysis.
Yeah. So I don't actually know. I don't really know how error works in a PCA kind of thing. The circle that is drawn is like the fitted oval that describes some level of consistency in the data, and if the ovals don't overlap, they're considered to be different. I think that's how that works, but I'm not 100% sure, honestly.
Yeah, well, in terms of so I also got some consulting on terms of this. Sorry, I thought we were talking about not the cluster analysis.
No worries.
That one. Yeah, the cluster analysis, it is normal for clusters to have a little bit of overlap, as long as they don't massively overlap.
Yeah.
So that's not necessarily an issue.
Yeah, I'm not really like a pro at this kind of data analysis. So when I'm not an expert in something, I usually just tend to go, okay, like I don't know enough to like fight you on this. I'll just accept that this is fine. Even though I agree, like orange and red, they look like they could, but they don't. So I'm just like, cool, yeah. And like nothing in biology is perfectly clean, right? I say this all the time, right? Biology is a spectrum. So even though they might look like they overlap, it would be really surprising if diet and exercise didn't affect each other at all, like 0% affect each other at all. Like that would be really surprising in biology. Biology is so interconnected amongst different systems that maybe there is some effect, but it's not significant enough to separate them in this cluster or to merge them in this cluster. So yeah, I tend to look at this and go, okay, cool, 4 different groups, sure. Yeah, a lot of variability in the high-fat diet plus exercise, which is also kind of interesting to note. Okay, so 4 different groups. Okay, so here is the orthogonal thing, right? So we find that exercise affects memory, kind of, diet affects anxiety, kind of, and then exercise is doing something to the microbiome, and then diet is doing something else, and they're mostly not related, whatever the heck they're doing to the microbiome. That's what we can get out of this paper. So that kind of explains the orthogonal title, and that's the main finding, I would say, of this paper, that diet and exercise, yeah, like the finding that affects behavior is not that surprising, right? Like that's just intuitively fine. The fact that diet and exercise don't affect the microbiome in the exact same way is, I think, somewhat surprising because biology is so interconnected that you would assume that they might have, you would assume that being healthy is going to affect your microbiome the same way, whether or not you do it through exercise or diet, but that appears to not be true. It appears that they have different contributions. Could the range of error be affected by subtle differences between individuals? So that's sort of the cop-out answer in biology in general, that there are individual differences between animals. What I will say is that Yes, it's probably a difference between individuals, but in biological systems, if you do random assignment and your mice are genetically identical, more or less, then those differences between individuals should be pretty small. So there might be something else at play for high fat diet plus exercise. It could be that small changes in the microbiome, or small changes, or yeah, let's spend a little time thinking about this because I think it's kind of interesting that you bring this up, Alice. So high fat diet plus exercise, it does appear to have a big spread. So I'm imagining that like high fat diet is like pulling the data along this dimension in one way, and then exercise is maybe pulling the data in the in like the down direction, right? So with two of these forces pulling on each other, maybe that's why there's more of a larger spread. You know, like metaphorically, like maybe because you apply two different treatments, that you get that spread, that larger spread. And you can see that non-fat diet, that normal diet plus exercise, yeah, I don't know. I like so my hypothesis would actually be that high fat diet is doing some weird immune things which can do weird things to the microbiome in very subtle ways that could have bigger effects depending on the initial starting condition of each mouse. So biology is messy, and I'm not too surprised. Okay. All right, so certain bacterial populations are correlated with anxiety behavior. So this is the big selling you supplements figure that grifters are going to love to cite, right? So there are certain bacterial populations that if you take the previous data and you plotted, you know, so this is the anxiety data, right? Time and light, and then the microbiome data. If you put that on the same axis, you see some vague sort of correlation where the more lactospiraceae you have, the more anxious you get, and you know, et cetera, et cetera. So like a really good grifter will take this study and go, oh, you know, I'm selling you a pill with lactococcus. See in panel E, lactococcus is negatively correlated with anxiety. You want to get rid of your anxiety? Take this lactococcus pill, right? So this is where you have to be really careful about interpreting data. And chat, hopefully I have trained you well enough to tell me why this data must be interpreted with extreme caution. Are the bacteria going to successfully take root if you introduce them into another system with different parameters for diet and exercise? So transferring microbiomes is certainly a thing. You can do a fecal transplant or a swab and gavage a mouse, so like stick the tube directly down their throat and pump their stomach with bacteria. The issue with microbiome studies, part of the reason why it's so hard to study, is that you can grow mice, you can raise mice that don't have gut microbiomes, completely sterile mice with no microbiomes. That's incredible. Born in completely sterile conditions, there's no bacteria that touches their insides. But they have a lot of health issues. And so while you can take like a bacterial population from one of these mice and propagate them into a mouse that has no microbiome, that's already problematic because you made a very, 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 very artificial mouse. A mouse that does not grow up with a microbiome is already going to be kind of a freak, you know, a kind of a low validity kind of freak. So you can do it. It could be informative, but you've introduced something that is very much not like a normal mouse, and that has to again be interpreted with extreme caution. Hairday says no causation was shown. Exactly. So maybe lactococcus is affected by something else that happens when you're not as stressed, and that's why they're appear to be thriving. Like you know by now how to not get got by grifters that want to separate you from your hard-earned money. How do they behave with no microbiome? They're a little weird. Yeah, microbiomeless mice, they act a little weird, I think. If I remember correctly, they definitely have like health issues. You need your microbiome. And so part of what is so hard about studying the microbiome is that there's no perfect control. There's no control that you can do that isn't going to be at least a little bit problematic, because we are born essentially from the moment that we are birthed with our microbiome. And there's just not a way to create a system that makes sense to have validity without sacrificing a lot. Hope that makes sense. All right. So any thoughts on this particular graph, Myelin? Or maybe I already took the soapboxing about grifting.
I mean, you took the soapbox for grifting. This is the set of graphs that inspired the conversation about effect size is the main thing that I can think of.
Because when I looked at this, I was like, they're kind of crappy. I don't know. Like, they're just like r squared of 0.26. That's like not great. Like, the green here looks like a straight line. So green is what condition again? Green is diet plus normal diet plus exercise. So here there doesn't appear to be any correlation. So overall, these r squared are very not inspiring to me personally. But yeah, you had a very different reaction, right? What was your reaction?
I literally have a note right here next to these graphs on this paper that says, those R squares aren't bad, with 2 exclamation points. So if that tells you the kind of like background that we're coming from and how we interpret this, we are interpreting those numbers from a very different lens.
I also really love that, because it just goes to show that like your microbiome is so complicated. It's as complicated as people are. Right? You could take that away from this, that like it's as complicated as the behavior of people, and that's kind of insane to me.
Yeah. I mean, for me, these are still decently good effect sizes, so maybe a bit less complicated behavior of people, or at least not as many, but it's still definitely quite complicated. There's a lot of things going on here besides the variables that they're looking at.
For sure. Do you know what FDR is? I actually didn't even know to look for that.
Yes, FDR is false discovery rate. So when you're doing a lot of tests, it raises your chance of getting a false positive, so thinking there's a correlation when there isn't. So when they say FDRP, that's a false discovery rate corrected p-value. It's saying we knew that we had a higher likelihood of null here than we perhaps should have, because we did so many tests, so we're raising our standards for rejecting the null. It's making it harder to reject the null. So this is a p-value where they essentially made it harder for you to reject the null.
So to walk through the descriptive statistics, and this is actually quite appropriate because I just taught the t-test today, the r squared, I guess we should have started with the r squared, right? The r squared value actually, do you want to explain it? Because I could do it, but I might butcher it.
I literally explained r squared in class yesterday.
Great.
R squared is the proportion of the variance in the dependent variable explained by the independent variable in a t-test. So the way that I like to describe it is, let's say in this context, if my r squared is 1, so 1.00, if I know exactly how many seconds these mice spent in the light, I would know exactly the relative abundance of, I can't pronounce anything, of that bacteria. It's the only, if r squared is exactly one, perfect correlation, the only thing that determines how much time spent in the light is the relative abundance. Well, determines implies causation, but it's a perfect relationship. Nothing else is influencing it whatsoever. R squared of 0 means there's no relationship. These things aren't related at all. R squared between is saying these things are related, and one of these things does explain the other. You can predict one of these things from the other, but there are other things. Oh no, my mic's cutting out. I'm sorry.
Seems to be okay now.
Oh, maybe I was leaning too far away. Sorry, but yeah, it's trying to describe how much of a relationship these two things have. Closer to 1 is a stronger relationship. Closer to 0 is a weaker relationship.
Yeah, and so if I could, so we have a question. What is FDRP? So hearing your explanation, let me try to say what I learned from you to make sure that I understand it.
Okay.
So FDR is false discovery rate, right?
Yes.
And so this is a way for the experimentalists to essentially analyze their data in a way that is statistically responsible.
Yes.
By increasing their standards, they are making it harder for themselves to find significance of this data. So R squared is not really related to whether or not the difference is meaningful, right? So R squared is like how much of the variance is described by the independent variable. A p-value will tell you whether or not that difference is actually meaningful. So it's a concept that a lot of students get confused, and I also have trouble with it sometimes. But FDR is the p-value after correction for this reanalysis of the data. Is that right?
Yeah, pretty much.
Okay, great. So we're still working with p less than 0.05 here, right? That's like their standard cutoff. So
yeah, essentially they've artificially increased the p-values that they originally calculated.
So if you have a p-value of less than 0.05 in these places, that implies or that suggests that this relationship is meaningful. So it might be messy from like a variance perspective, like, oh, only 24% of the variance is explained by independent variable. That would be a very weak relationship. But even if it is a somewhat weak relationship, it is meaningful. It's two different ideas, right? Meaningfulness or realness and magnitude. So two different descriptive statistics to think about.
Yeah. I think of it like a metal detector. The p-value will only tell you if there's something there or not. The R squared value tells you exactly how big the thing is.
Oh, I like that a lot, actually. If you want to learn more about statistics, Myelin has a whole stream series of an introductory to stats that you can check out on their YouTube channel, linked in the description.
Oh, thank you.
Always be plugging. Okay. Yeah, so again, like grifters will try to sell you on this, but like being aware of that, it makes it okay to interpret this data. It looks like these relationships are meaningful. So it looks like we do have a relationship between certain bacteria and anxiety, but as many of you smart chatters have pointed out, it does not appear to be causative. And it would be kind of hard to determine if these are causative, just because the microbiome is so messy. so messy. And, it would be tempting to think, oh, let's grow a mouse with no microbiome, take a lactococcus and inoculate that mouse and see what happens. But the thing is that would never happen in a real organism, just because the microbiome is a community. There are a lot of microbes in the gut that cannot be cultured outside of the stomach, or outside of the gut, I should say. And so it's just not a realistic you would not be able to draw conclusions that would matter in a real person if you were to do that experiment, which is why these microbiome studies tend to rely quite heavily on correlation, and that's why progress, I think, is kind of slow. And it's not a fault of the microbiome people. The microbiome itself is what's causing these issues. Why can't they be cultured outside the gut? That's an excellent question, Alice. So there is like a dark microbiome of unculturable bacteria that need other bacteria to survive. So say that you spent billions of years evolving, with your best friends, Streptococcus pyogenes. I don't even know if that's a real bacteria, but say you have a best buddy that you co-evolved with your entire life. Now, over millions of years of evolution, you can no longer survive without the exact formula of things that bacteria is making for you. So bacteria are very complex in their communal relationships. You have to be like a very strong, independent bacteria to survive outside of the microbiome. Like E. coli is a fantastic, ubiquitous bacteria that is not only a part of your microbiome, but also it's just like everywhere, right? But those bacteria have evolved to be very generalist with their survival kits. Shadow microbiome, yeah. Very dark microbiome, I think is what they call it. we should learn from the guys trying to save ecosystems by introducing other animals that do the exact opposite. Yeah, we can't really change the microbiome in a research relevant way without, like, it's hard. I'm not saying it's impossible or that science is bad, but again, you have to interpret those kinds of things with extreme caution. Just because the interactions between, like, if you increase the population of one bacteria, you're also potentially decreasing the other bacteria. Now, is an effect because you increase this bacteria or because you decrease those bacteria? Like, those things are just some of the questions that microbiome people have to tackle. Okay, so complicated stuff. So other populations are associated with contextual fear memory. So that previous slide was about anxiety. This is about the foot shock. So, freezing context. And again, even worse, R squared, 0.11, 0.1, like very, like not a 0.09 with a p-value of 0.0489, just hovering around that cutoff. And these don't appear to be corrected. I wonder why, right? There's no FDR here.
Yeah, their use of FDR is inconsistent.
I wonder why. I actually don't know why. It goes a little beyond my expertise.
I don't know why either.
Yeah.
It could be they had some rule where above they're doing 6 tests, so they did FDR, but here they're only doing 4, so maybe if it's 5 or more tests or 6 or more tests, they'll do FDR. But I don't believe they did FDR on some other things where they did more than that, so I'm honestly not sure.
Yeah, microbiome stuff also includes a lot of statistics that I'm not familiar with, which is also why I tend to stray away from covering these as journal club articles, because I get to show my whole ass and like not understanding statistics. That's why I'm like, oh, actually this is really good that Myelin is here, because I like don't understand these statistics. I would have just brushed past them and be like, yeah, that makes sense, and I would have moved on with my life.
I mean, given that I'm not familiar with the norms of statistics and biology, I'm not sure how helpful I'll be, but I'll do my best.
But the thing is that norms are themselves a sociological construct. Oh, we can get into it. We can get into it after we're done with the data. After we're done with the data, we'll get into the. Yes. All right. So conclusions, pre-scrutiny. So just reporting what I understand to be the main conclusions in this paper. 1, high-fat diet in mice increases what looks like anxiety. 2, exercise improves contextual fear learning, probably in this foot shock model, although there are overlaps with anxiety. Diet and exercise have orthogonal effects on the microbiome. So diet affects the microbiome in one way, and exercise affects the microbiome in another. There are certain bacterial populations that are kind of correlated with anxiety and memory, although the strength is not high. That's not surprising, right? Like, behavior is so complex. It would be really surprising if 100% of your behavior were controlled by your microbiome. It kind of implies that you're like a little Gundam, and the Gundam pilot is the bacteria in your gut. That's probably not true, so we shouldn't expect the correlation to be very high. But it's there, and it appears to be statistically meaningful, so that's interesting, although we don't know which bacteria are causative, which one are correlative, for future research to determine. Oh, there's a question that Harry has that I have recently learned what preloading means.
I do not know what it means.
Oh, now I'm not sure if I know what it means. I think Cideray was talking about it on stream earlier this week about like declaring your hypotheses. Declaring your
pre-registration.
Pre-registration. Yeah.
Mm-hmm.
I don't know if that's preloading. I don't know if that's the same as preloading.
I've never heard it referred to as preloading, so I don't think so. I'm fairly, at least I used to be fairly involved in the community pushing for pre-registration, and I've never heard that language, so I don't know. Yeah,
Harry, you might have to elaborate, because my head went to pre-registration, which I didn't know about before. That's pretty interesting. Mm-hmm. Okay, so let's talk cross-cultural collaboration. So I think these are the big findings of the paper. Again, in terms of like what I have to say, I wish they didn't do the foot shock. I wish they did novel object recognition instead, but that's really my only complaint. Everything else I think seemed pretty all right to me, not looking too carefully at the statistics. but you actually have a bit of a different perspective coming from a different field. So you are not familiar or biased by our norms. So it's kind of interesting to get a read for how you reacted to the paper in particular. But before we move on, let's just kind of address some of the chat. Can you explain in like the more official language, because this is just me watching Side Array's stream, about pre-registration?
Sure. So this goes back into history. History back in 2011. So there was this big crisis in social psychology where we found that a lot of our reading.
Myelin, your mic is cutting out again. And you might need to be a little closer to the gate.
Hello.
Yep.
Okay. Sorry about that. Yeah, so there's this big crisis in 2011 where we were concerned that a lot of findings were not replicating. And one of our concerns was something that you actually call researcher degrees of freedom, all choices that we make for designing our studies and analyzing data. And so we were concerned that we were getting findings that weren't real because people were changing their plans while they analyzed their data or collected their data. And so what we did was we invented this thing. Well, I don't know who came up with it, but there's this thing called pre-registration where we write down all of the things that we're going to do in as much detail as we possibly can, and then we post it on a website where we cannot edit it. My mic hates me, I'm sorry. Yeah, so pre-registration is posting your plan where you can't edit it so that people know exactly what you intended to do.
Yeah, on Side Array's stream, I heard pre-registration referred to as like when the anime character declares their attack.
Yes, I love that metaphor. Yes, I think that's a great metaphor.
It's like Joseph Joestar in part 2 of JoJo's Bizarre Adventure has this thing where he can like predict the future, and he goes like, "You're gonna attack me, aren't you?" Like, and he'll just like predict what they do. It's kind of like that, stating the intention so that you can't look at the data afterwards and be like, "Ah, this was my plan all along," right? So this is a little keeping psychologists a little more honest. Although I don't know if that's a common practice in biology.
I'm not sure. And this paper was published in 2014, and that was only when pre-registration was really starting to catch on in psychology. And I think these sort of practices haven't pollinated to most other places. They really got popular in psychology around 2012, 2013. So I can't imagine that biologists were doing it yet at that point.
What is interesting, though, is that in biology, we might not have pre-registration, but we do have places in our papers where we can talk about unexpected events. So even if we're wrong with our hypothesis, we're usually pretty honest about things not turning out the way we expected them to. I don't know if psychology had that, or because we say like unexpected, contrary to our hypothesis, X, Y, and Z happened. So that's like our version of that.
Oh, well, like, yeah, we definitely say stuff. Oh, there's no like dedicated section in our papers for that, but we definitely say stuff like that. But I could do a whole video on all the stuff. It's complicated because you've mentioned telling a story earlier. And for a long time, a lot of the advice that we had writing papers was to make them more understandable. You need to tell a story. And sometimes that means claiming that you hypothesize the results correctly because it makes it easier to read the paper, and it's a whole thing.
I actually find that my favorite papers are ones where they admit that they were wrong initially. I just find them to have a little more drama to them.
Those are very fun, but sometimes they can be confusing to read as well.
Yeah, they definitely can be, and a lot of papers in biology are quite. I think of the sciences, I think I would rate biology to be amongst the easier to get into, just in terms of understanding the base conclusions. Probably just as hard as other disciplines in terms of actually getting everything, but entry level, I think it's easier.
Could be.
I think
Could be.
psychology might be pretty similar, where like you could get in on the ground floor and like get the broad strokes pretty easily, but like understanding the nuances where that's why you go to school.
Yeah, definitely. Definitely. I think it's also easier in psychology to trick yourself into thinking you know what's going on.
I think that's also true. Yeah, for sure. Because I've read a couple of psychology papers. I wanted to be, I don't know if you know, but I wanted to be a professional psychology person.
Oh, that's so cool. I did not know that.
Yeah, I was in my senior year of undergraduate deciding whether or not to study the sociology of memes and or online radicalization or molecular biology. And I was like, well, one of these on average pays better.
I took the less paying one, so
yeah. That is to say, I do share a great kinship with you know, you psychology VTubers, just because like, you know, it's so cool to talk to people who I could have had a similar life to you in a different like universe, but I went with a different path because I was also like very deeply interested in conspiracy theory and all that kind of good stuff.
I still think it's wild that there are two conspiracy theory psychologists who went the VTuber path. That is amazing to me.
Yeah, in terms of the p-value on that that cannot have been an accident. It would be too crazy for that to have been an accident.
Well, we can never prove anything. There's always a small probability it was an accident, but I do suspect that there probably is some variable there.
Yeah, right. Like some explanatory model that allows you to that pushes you to do that kind of thing.
Yeah, absolutely.
All right. So, let's talk a little bit about cross-cultural collaboration. Getting your perspective. So from my perspective, this is like kind of out of my field because I'm a lot more of I'm a salt and potato biologist. I like my Western blots and my qPCRs and things that you have experimental control over. I'm way less comfortable in the realm of correlation because you have to know a lot more math to be able to figure out whether or not things mean anything. So I typically stray away from these. So I would actually say that this is sort of in between our fields, more on mine than in yours. But methodologically, in terms of statistical analysis, you actually are coming in from a related field because you deal with correlation so much. So I kind of wanted to get a sense for how you reacted to reading this paper. But before I do that, I just added this slide in at the last minute to explain to you my full appreciation for this beautiful title. And I say that this title is beautiful for a reason, because when you are in graduate school, you get told that you are wrong like all of the time, and you start looking for ways that other people are wrong. And that's just a part of being an academic, right? You get roasted constantly. If you're ever even a little wrong, you get actually. Your entire life is actually the moment you step into graduate school. And so this is one of the best titles I've ever seen because you actually can't actually this. So diet and exercise. So notice it doesn't say a high-fat diet because we don't know if it's high-fat diet. Actually, this entire thing we've been calling it high-fat diet, but we don't even know if high-fat diet did this. What we know is that 60% lard did this, right? So the fact that it didn't say high fat leads to increased anxiety, which I think is a reasonable conclusion to make from the paper, you can get actually there because maybe this isn't about fat, maybe this is about lard. So that's already beautiful. Just the first word is already great. Like a very careful title in the fact that they say diet, which is true. They changed the diet, and a change in diet did affect anxiety, did affect memory, right? Like that is a very careful choice to not include high-fat diet in the title. And exercise orthogonally. So this is a bit of a jargony word that I don't love, but basically means unrelated effects, alter the gut microbiome, and reveal independent associations, right? Because you can't call them causative in a certain way with anxiety and cognition, right? So it's a very, very, very careful title that explains exactly what the conclusions are. Changing diet and changing exercise regimens is related to changes in anxiety and cognition. Although technically I could actually the anxiety thing is, who knows if that's actually anxiety, but it looks like anxiety, so I'm actually okay with that. But I really do like the choice to not, because you can imagine the grifters, the health grifters in this world. If they see the title "high fat diet increases anxiety," bang, you get a whole generation of grifters advocating for low fat diet, which might not be the worst thing, but they might not be doing it under the best pretenses, right? This is a very careful communication of the actual results. Yeah. So let's go back then to your understanding of the paper. And first, before that, we'll just answer some of the questions that came up in chat. So, Myelin, do you want to explain what your field is one more time?
Yes. I am a social psychologist. So I study the psychology of how people behave and think and feel differently in the presence or imagined presence of other people. And my specific thing is how context predicts belief in conspiracy theories.
Super, super interesting. Again, the kind of research that I was really, really into when I was in my senior year of undergraduate. There's something almost biological about how conspiracy theories spread, like viruses. It's like it combines like a lot of my academic passions. It's pretty interesting.
It's really neat. And I think rumor spreading and fake news spreading does absolutely use some of the same statistical models as, say, epidemiology, which is wild but cool.
Yeah, that's why I'm like, I love biology, because it sits at the center of so many other disciplines, that there are so many overlaps that you can have with other fields, and like there's something very appealing about that narrative power. High fat diet causing anxiety is a dangerous title for issues related to fat phobia. Yeah, I mean, that's definitely true as well. Part of exactly why I like that they didn't specify what kind of diet, but rather generalized it strategically to avoid those associations. I don't know exactly what their motivation was to do it, but I know that it worked very well for me in particular. Okay, so this slide is one that you actually made, Myelin. Coming in from your field, you took a look at this graph in panel B, and this did not work for you. Can you explain why, and then what you changed it to, which is to the right over here? And I'll actually move us out of the way so that we can see your version of their data, because I think this is really, really funny.
I do too. So when I looked at the graph on the left, I had a lot of trouble understanding it. It's very different from how people in my field would normally organize a graph for this type of data. And I think we discussed it's probably because we're interested in different parts of this analysis. But what they did here was something called a factorial ANOVA. And they say this in the note. They did 2 independent variables on 1 dependent variable. And I'm used to looking at this as like, did exercise change the effect of diet? And so typically we'd portray this as, here are the 2 control diet columns, here are the 2 high-fat diet columns. And then you can see how the effect of exercise differs depending on diet. And that's how I'm used to looking at these graphs. So when I saw the one on the left, 4 different conditions, it messed me up something bad. I could not understand what was going on, and I remake the graph in the way that I was used to looking at to figure out what was happening. And looking at the effects that they have here, I think of this as essentially 3 different statistical tests. There's first asking, does diet have an effect on anxiety? And in this case, time in the light, higher numbers mean less anxiety. So I would ask, are the bars on the left on average higher than the bars on the right? And yeah, that's what they find. And you can see this in their graph. It's a bit harder for someone like me to see, but on average, both times the control diet bars are higher than the high-fat diet bars. And then the second question would be, did exercise affect anxiety? And I'd be looking at, on average, are the lighter bars higher than the darker bars, or something along those lines? And then their interaction, is the difference between the exercise bars, is it different depending on the diet? So I don't even know what to say other than that I had a very difficult time trying to figure out all of those things from the way they organize things on the left, because it's very much not how I'm used to looking at this, not how we are trained to look at these types of graphs and interpret them.
That's really
I had a
interesting.
wild time.
I really, really love that you did this, because I like, until you explained it to me just now on stream live, I had no idea what your graph meant. I was like, it's the same data, like I know that, but like me looking at your graph, I was like, I don't understand what I'm looking at. It's really hard for me to interpret the way that you put out the data, which is a really fun little like difference, because as a biologist, the design on the left is, from my experience, a lot more typical, because this makes it easier to examine the difference in treatment groups one condition at a time, right? So first, you see the normal diet, which is the control, like total control, no treatment, essentially diet. And then the next question you want to ask is, okay, what is the effect of a high fat diet? So you put the high fat diet next to the normal diet, and you see that decrease. Great. What about exercise? Does exercise affect it? Then you look at the green. What about high fat and exercise? So this is laid out almost in the order that you would ask experimental questions. And so that's the way that my brain works. I get why you plotted it in your way, and I think it is in ways cleaner from a statistical analysis perspective, but it's a little harder to wrap my head around in terms of comparing conditions, like in the order that I want my questions to be answered, if that kind of makes sense.
Yes. Yeah, it seems like you're more interested in what we call the main effect, so just each independent variable on its own. Psychology a lot of the time is asking about interactions, so how does one independent influence the other independent variable? And I think we're doing a poll.
Yeah, I'm just kind of curious, chat. Which of these two graphs do you prefer visually?
When it doesn't go my way, I'm going to blame sampling bias. Yeah, so I think it's just the different goals and what we're interested in. Like, it seems like you and perhaps other biologists are more interested in the individual effects of the independent variables, and I'm coming from a perspective.
The left one is labeled B. Yeah, that's confusing.
Oh, that's confusing.
The graph on the left I'll call A because it's on the left, and the graph that Myelin made we'll call B. Sorry about that. Sorry. Sorry, you were saying, Myelin?
Oh, I just think it's because we're interested in different questions, and like your setup for the graph makes a heck of a lot of sense for the way that you're thinking about it, and my setup for the graph makes more sense of like I'm interested in the effect of these two things. Like, do they do something different together than they would have separately? So it's the goals, the goals that we have when looking at the graph are different, which means that we think one graph makes more sense than the other for our particular purposes.
And I think that's a really fascinating difference, because I would, like, in no universe think to show the data like you showed it. But hearing your explanation, I'm like, we should maybe do that more. Although I don't know, like trying to do the operation that I did on the left-hand side, thinking like no diet versus high-fat diet, that actually gets kind of hard to do looking at your graph.
Yes.
Like I can trace my finger and point to what I need to point to, but it's actually a little visually confusing to do it that way, in the order that you have the graphs, even though I can definitely see the utility of doing your comparisons that way. Yeah, because again, since you are more familiar with the statistics that are that underlie this makes more sense when you're looking at interaction versus a 4-cell design, like treating each experimental condition as its own unique group versus 2 axes of manipulation.
Yeah, I and it is difficult to like I'm really used to it, but when I teach it in class, there is absolutely a lot of drawing on graphs, and like you're essentially in your brain turning this graph into one that looks like this or what. It's not an intuitive skill. It's very difficult to learn.
Yeah, I can imagine, because I'm like, these are the same words, it's the same data, but why am I having so much trouble like reading this? It's actually really interesting to know that we have trained to look at the data in different ways. It's pretty interesting.
Mm-hmm. Yeah, it is weird.
All right.
Oh man.
So it actually looks like chat prefers your graph over the graph that
I was
so
not expecting that, but I'll take that victory.
I am going to claim sample bias now.
There is some data. I didn't know what their error bars were, what numbers were going there, so I couldn't make it perfect.
Yeah, but that's kind of cool. I will still prefer left more than right because I can more easily critique the experiment if I go from left to right in the order that I want my questions answered. But I can see that like this interaction here, you do sort of lose this point, like the intergroup comparison. You don't really get that from the left panel. So very, very
Different
cool.
goals.
All right. So you also had some things to say about the way that the authors did statistics, which I found to be very fun to listen to. And this was also the point I was like, oh, this psychologist is going to see how we live in biology. Oh, no.
Well, I mean, I really don't want to sound too critical because I know that there's a lot of different considerations y'all have. I'm working with human participants, and y'all are working with mice that are specifically bred to have similar genetics, and mice and human, like there could be very good reasons that we do things differently, and it's hard for me to say. Like this first one you pointed out, there's a very good reason first of all, so central limit theorem.
Yes, let us start with the central limit theorem, please.
Let us start with the good old central limit theorem. So the central limit theorem is guidance on how many participants or like observations we should have per cell or per condition. And the idea is we're using samples. So we're using these, say, there was 10 mice per cell here, 10 mice per condition. And we're using those 10 mice to try and generalize population of all mice who could have potentially been in that condition. The central limit theorem says that once we get to a sample size of 30 observations, we can be reasonably sure that our sample population mean. We can be reasonably sure that we're getting a pretty decent estimate of the population. It's not going to be perfect, but it should be okay. So we would like to see a minimum of 30 observations per condition. So with a design like this, with 4 cells, or you can think of it as 4 conditions, psychologists would want a minimum of 120 people, or 120 mice total. And this study has 40, which I was concerned about, but I think you make a very good point that, first of all, they have tried very hard to make as big of an effect as possible with that 60% by lard diet. And there are time constraints. There's financial constraints here as well. And then also, in terms of we don't want to harm our animals than is strictly necessary. And this happens in psychology too, and especially like the neuroscience things, brain scans, those are expensive. You can't afford to have 30 condition all the time. So I am concerned about sample size, but reality is a harsh mistress.
Yeah, and so sort of cushion the difference, like central limit theorem, n equals 30, those assumptions matter for assuming that you have a normally distributed population. Is that correct for the central limit theorem?
To an extent, there's some theoretical reasons why the central limit theorem is based on a distribution of sample means, which theoretically should always end up normal. But of course, we're talking about scores here. I have problems with the central limit theorem. I think it makes some logical leaps that I think you're hitting on right now.
In any case, what I do know in terms of like the little baby statistics that I'm aware of, when you do a t-test in particular, you don't have to have a very high n, but you use a different distribution that isn't the normal distribution. You use the t distribution, which works for smaller n's. So it doesn't necessarily mean that it's like great to have small n's. It just means there is a specific test that allows you to work with smaller n's, and that would be the t-test.
The t distribution is a normal distribution.
Right, it approaches normal as sample size approaches infinity, right? That's how I understand the t distribution.
Yeah. It's normal in that it's unimodal and is symmetrical. The t distribution is just a distribution, it's a series of are adjusted based on the sample size that you have.
Gotcha.
Yeah, it's
tricky.
super weird. Yes, very, very tricky.
To give you all an idea, and why, like, if you see angry Redditors be like, oh, this study only used like 7 mice or whatever, to give you an idea of why that's reality, because I see a lot of Redditors dismiss mouse studies out of pocket because of low ends. To give you an idea of how expensive this is, it's $1 a day to keep a mouse alive, a cage of mice alive. So with an N of 120, each mouse cage is about 5 mice is how much you can house in a single cage. So some quick maths, that's 24 cages. So that's $24 a day for a study period was like 20 weeks. It's expensive, right? And that's just to, have the mice be alive. That's not even accounting for paying the researchers or the husbandry staff, et cetera. So yeah, you don't want to use more mice than you have to. You don't want to spend more money than you have to. You also might not need to use that many because you can do power analyses, although that itself can be quite problematic. But that's maybe a discussion for another maybe we could talk about it later too. I don't know.
Yeah,
If you're feeling up for it, you can get spicy with your thoughts on power analyses. Okay, and then you had mentioned that they were following up non-significant events, increasing the chance of false positives, but they do use post hoc corrections. Can you explain a little bit about that?
Oh boy, yeah. So the type of test that they use a lot in this is a factorial ANOVA. And one of the parts of a factorial ANOVA is an interaction, which is essentially testing. Oh man, how do I put this? The interaction is a kind of complex thing where it's how 2 variables work together. And if the interaction's significant, that means that there's some difference between your 4 cells. And you can go and you can look for those differences between those 4 cells. If your interaction is not significant, that means there is no meaningful difference between those 4 cells, and you should stop. You shouldn't look for differences because it's already told you there isn't anything here, or there probably isn't anything here. In this paper, on a fair number of occasions, there's a particular table where they do it, I think, a bunch of times. They follow up on non-significant interactions, and that first interaction test is supposed to be kind of a gatekeeper. It's saying, if this is significant, it's safe to go forward. If it's not significant, beyond here be dragons. You might get significant results, but those significant results are probably false positives. They're probably just a statistical fluke, and they probably don't mean anything.
Yeah.
But they go in, yeah.
So I'm just I think this is really valuable coming from you, considering a lot of your field is disentangling what is real and what is not. Again, with the whole replication crisis issue, like, I hear you say that, I'm like, that is coming from someone who has been in this field where you do find a lot of false positives, and that contributes to people not being able to replicate the research. So this idea of chasing these threads that don't deserve to be chased, that's kind of how it feels, right? Like they're chasing things that they don't have to be chasing, or they shouldn't be chasing because they're non-significant.
Yeah, and if they do end up being non-significant, or, well, if this thing isn't actually real, if it was a false positive, and other researchers read this paper and design entire studies of that, they are sacrificing tens of mice to something that isn't real.
And months.
And could have been avoided. And months.
Hundreds of thousands of dollars.
Yes, yeah. That's a lot of stuff down the drain because somebody followed up something that they shouldn't have.
So
and then if that gets implemented, no, sorry.
So in psychology, there's a big faux pas then, right?
Yes, this kind of thing could get you, and probably on multiple occasions has gotten people rejected from journals. You wouldn't be able to publish it if they interpreted those results. So they said, this has meaning, and this is what we think it means. Oh, that would be really bad. I have seen people, particularly spiteful people, do some really horrible things to people who have done this. Career-wise, critique-wise.
Yeah, for sure. Gonna have to go soon. Thanks for stopping by. Thanks for
Yes, thank
sitting
you.
through our talk on the microbiome. Yeah. So I don't really have an opinion on following up on non-significant effects, just because, again, this is not the kind of research that I do or I did in graduate school. So having heard your logic, I totally agree with you. It makes a lot of sense. I wonder why the authors didn't. Maybe it's just not like a cultural thing, because here's the deal. Chat, here's the dirty little secret, is that Myelin has a rigorous statistics education and I don't. And this is something that I've always been kind of insecure about, just as a scientist, right? Just as a scientist in general. And you could say, well, that's because you went to like a bad school. I went to like one of the best biology programs in the States. I don't say that to brag. I say that like even at the highest level of biology education in the US, we're not getting a strict, relatively controlled statistics learning experience, right? The only stats that I had formally was 101 in undergraduate, where you learn t-test and ANOVA and, you know, X, Y, and Z tests for very, very simple experiments. But when you get to be a working scientist, right, your experiments are not often as simple as X group got Y treatment compare with Z group that got no treatment, right? They're not always that simple. And so you might have to do all sorts of statistical tricks in order to, not tricks, I shouldn't call them tricks because that's demeaning.
You can call them tricks.
Some statistical methods, I should say, to appropriately, I'm not going to say to hack your data, but to appropriately and responsibly interpret your data, right? Like there's a whole world of statistics that goes beyond 101 on how to translate what you learned from 101 into reality. And that is not something that I got as a formalized part of my education. I got the t-test, I got ANOVA, I got a couple other like tests that every statistics 101 student knows about, but then the real world is often a lot more complicated than just running a t-test. Like all of these things about corrections, following up on non-significant events, etc., those are not practices that were rigorously installed in us. Instead, what we had to do was rely on older members of the lab to tell us what to do, right? Like unless you were yourself very motivated and driven to learn statistics on your own, you were just going to inherit whatever statistical methods someone before you was doing. So I think statistical practice gets inherited through lab culture rather than something that is more centralized and disciplined. I don't have any evidence to show that that is true, but in my personal experience as a graduate school in a very good molecular biology program in the States, that is what seems to be happening. That unless you go out of your way to inquire about statistics or collaborate with a statistician or a biostatistician, you are pretty much just following along with what other people are doing, which is in contrast to you, right, Myelin? Like in graduate school, you still had to take stats, or right at some point?
Yeah, I would say the program that I was in does have a bigger stats focus than a lot of other programs. We have specifically an area in it with dedicated faculty that are all about statistics. But we are required to take at least a year or a year and a half worth of stats classes, and we had the opportunity to actually minor in statistics, which is what I did.
But as a graduate student or an undergraduate?
Yeah, as a graduate student.
Oh, wow. I didn't even know you could minor as a graduate student.
They got rid of that at my program, but it was really cool. But yeah, I think psychology takes statistics very seriously. I think part of that is our insecurity when people call us soft science.
Bullies.
And yeah, I think another part of it, though, is like you mentioned earlier, we're dealing with some really complicated stuff, and we've got correlational stuff going on. And like in social psychology, there is differences between areas of psychology, like neuroscience as concerned, I would say. But we have to worry about a lot of confounding variables, and we're studying people that are inside situations, that are inside cultures, and there's a lot of, we need some really complicated stuff to do what we're doing.
Yeah, because if we compare just our experimental systems here, right, like you're dealing with whoever you can recruit, right, which may not be a randomized sample. And then you probably have to do all sorts of statistical judo to actually make that work. Whereas for a biologist, you have genetically identical mice, more or less, that you control their diet, you control their sleep cycle, you control their exercise, you control everything in that mouse's experience, which means that the statistics that we generally have to do are a lot less complicated than for the mess that is whoever you can get.
Yeah, and apparently I did talk to one of the statistics faculty, and they did say that, and maybe it's because of all this control that you have, but apparently mice do have less variability on measures than human beings. And it could be because of all those controls. It could be because maybe human society is more complex than mouse society. I don't want to assume, but it does sound like there are statistical differences in the very data that we're working with, besides the type of hypotheses we have to deal with.
For sure, for sure. Yeah. Do you want to say about say anything about the post hoc corrections? Because I thought that was also interesting as well.
Yeah, that was the FDR stuff, the false discovery rate stuff. And yeah, the weird thing is they only use the FDR for very specific stuff, and I don't remember if they mentioned if they used it when they were following up these non-significant interactions. But it is good practice, and that surprised me that they were following up on those non-significant effects, which is like something I teach my Psych 1, or like my stats, intro to stats students not to do. Like, I really emphasize it. Something that psychology undergraduates should know. But then they were doing post hoc corrections, which is something that, like, you will find published papers that do not use post hoc corrections when they really should. So I don't know. It was very interesting.
My feeling is that maybe they got a consult from a statistician and they just did whatever the statistician said.
I think that's probably the case.
It could be so. Although microbiome people definitely do tend to be better statisticians than some of the other more, meat and potatoes molecular biology labs. Just because, again, their hypotheses are just as complex as people, because the microbiome is crazy complicated. And the more complicated your system gets, I think, the harder your statistical analysis will necessitate you having.
Oh, absolutely.
Yeah, I remember in a previous life I was I had emailed a psychology professor about their social science data about scientific reasoning. And I had all sorts of questions about like what counted as a strong effect, because it was just such an unknown field to me, where like, is like an R squared of like 0.2, is that like good? Okay. But like there was like a whole section that I just didn't understand, all the corrections and all the statistics trying to normalize for all these cofactors, covariables, just because people, everyone's unique, and that makes it very hard to make generalizations about data.
Yep.
Yeah, but that's the part where I was like, oh god, they're gonna find out how we live. This thing about following up non-significant effects, I can't imagine an argument for doing that the way that you explained it. It might be that we're just not as good of statisticians as psychologists are, because even in like my graduate program, I talked to the psychologists there as well, and they were all TAing like freshman statistics courses, because you have to be good at stats to be a psychologist. It's like a requirement.
Yeah, you'd think people would avoid that, but I have grad students vying to TA for my stats class. That's like seen as a very desirable thing to do. And also it, of course, increases your marketability and salary and job prospects if you're good at stats. For
sure. So recommendation for those undergraduates out there: get good at math. Math is really good.
You don't have to get good at math. Just get good at stats.
Yeah, get good at stats. But also, if you can get good at math, get good at math.
That would be a good idea too. Yeah, but generally. I didn't need to.
I didn't need to either, but like, if you get good at math, the world becomes a lot easier, I think. I think getting good at math is a quick hack at certain types of success.
Probably so.
All right. So you actually went above and beyond in terms of a collab here, and you actually searched the literature for the equivalent data in humans, which I found to be a very, what's the word that I'm looking for? I guess I'm amazed. I wouldn't have even tried. I wouldn't even know where to begin or even how to do that. So can you explain how you generated this data?
Well, first I tried to contact the original author of the paper because I wanted to see their data because I
That's
was
already
just
crazy.
unused to it. Well, the thing, I really wanted to know, and the original author's email has long since been deactivated. I think they retired. I hope better place. But so I went looking for existing data sets that had information on exercise, diet, and anxiety. And of course, I was very limited by what people had done. So a lot of this data isn't exactly comparable to the variables that they were using. And of course, I have nothing on microbiome. But I did find some CDC data. Unfortunately, these were 3 different data sets, so there were very few exactly participants who had all the data between the 2. The x-axis of this graph is exercise time. I believe this was how many times they exercise or something along those lines. Lower numbers, of course, is less exercise. Higher numbers is more exercise. The y-axis is self-reported anxiety on a scale of 4, I think. And then the different lines are their self-reported weight. So this is self-report, and it's only 3 categories. It doesn't necessarily say anything about diet. They did have diet data, but only for children, which was unfortunate. So the blue line is underweight, and clearly they did not have a lot of data on that. Crazy things, and the only people who were underweight, they had no people who were underweight who were times than like 6 times a month. So lots of missing data there. For overweight people, their exercise was not related to their anxiety. For people who reported being about right, I believe their more exercise was related to less anxiety. I should have pulled up whether these effects were significant after the fact, but it is a pretty small sample, unfortunately.
Yeah.
Oh yeah, if you're not, yeah.
So if we wanted to compare this to the mice, we actually saw that high-fat diet increased anxiety, but exercise didn't. So this data, I mean, yeah, with the N of 40 different, you essentially compiled this into sort of a pseudo meta-analysis, which I think is really fun. It can't really say much about the underweight people, even though like this is like such a severe decrease.
Well,
you said that this was like a very small sample. What did you say this was?
yeah. Oh shoot. You know what? I should just pull up the analysis right now and find out. Because it looks like there's, just looking at that, it looks like there's maybe 2 people in there. I think it was more than that. But very, very small sample size.
Gotcha. So might not be super applicable, but this again just goes to show like how complicated analyzing human data is, right? Not only is this all self-report, so you know you have to take it with a mountain of salt, but yeah, just you can't prescribe exercise as easily as you can to a mouse who has to run on that wheel. Marlon, you see 4 blue points. Yeah, maybe.
Let's see. I'm almost there. Summary as factor. Sorry, I gotta write some code.
No worries. Yeah, it's crazy that Myelin did this anyway. Like when they sent me these slides, I was like, "You did what? how did you even get the data?" Like that's bonkers.
I'm sorry. Also, it's past my cat's dinner time, so if I suddenly disappear, it's because he's eaten me. Oh gosh. Okay. Oh. It hates me. Never mind.
So,
I'll figure it out eventually.
No worries. Akasa Adrian says you don't know how many times I spaced out during statistics lectures. The thing about being good at statistics is that makes you the ultimate actually person. That is a superpower that you get if you pay attention in stats and you get good at it. You can become the final actually boss. And that's pretty appealing, I think.
Oh, well, if you get good enough at stats, then you realize you don't really have a right to actually anyone.
Maybe. Yeah, so this was a valiant effort, definitely, to try to see whether or not the human data align with the mice data. And I can probably say that this is probably not very conclusive at the very least.
Oh, definitely not, no.
Okay. But then you did not stop there. You actually went and did something else. So can you please explain this data?
Aha! Ah, yes. So this data was from a much less reliable source than this. Um, I wanted a bigger sample size. So this data is from a website called Kaggle that compiles data sets for practice on like machine learning and data analysis, data science. So it's not very clear where this data came from. It seems like it was combined from a bunch of different data sources. So I don't know how reliable it was. Over 7,000. It was 7,022 to be exact. Again, this is all self-report data, and I graphed this in the way that I would graph interactions, so I'm sorry if this is hard to interpret if you're not used to it. I did not find anything here, though. So the x-axis is diet quality grouped into poor, average, and good. This is self-report, so who knows if they're honest. The y-axis is anxiety score from 1 to 5. Higher numbers, more anxiety. The red bars are low physical activity, green are moderate, and I did not find there. Diet did not predict anxiety, physical activity did not predict anxiety, and they didn't interact to predict anxiety here.
Yeah, so this isn't exactly one-to-one with the mice experiments because this is just diet quality. There's no like specific high fat diet hypothesis here because it could be specifically high fat is doing all these terrible things, and diet quality can mean anything to, anything from tons of fat to tons of carbs to tons of sugars, or maybe you're not eating like your micronutrients, maybe you're not eating leafy greens or something. Like, it's hard to know what diet quality really means. So this is not necessarily disproving the mice result in humans. It's just we can't really learn anything additional by looking at this 7,000 self-report data. Yeah, so it's something that would have been cool if it did align, but because it doesn't align, it doesn't actually prove or disprove either. Yeah, it doesn't really tell us much here. Yeah, so it was a very cool attempt that you went out and like did it. But again, like, I think if we're interpreting our mice data with extreme caution, this should be interpreted with extreme caution.
Yes, like multiple extreme caution.
Yeah, and self-report data is not exactly very reliable. But again, like for psychologists, that's a problem for you all because that is just what you can do sometimes. For biologists, self-report, the mouse isn't self-reporting anything. We're measuring its actual behavior. It's an advantage that we can get, but again, we lose out on the fact that it's a mouse. So there's this like eternal tension of internal and external validity. And if you are thinking about what to specialize in, if you're an undergraduate, you kind of have to find where your sweet spot is. So I like control. I don't know what that says about me as a human being, but like, ultimately, even though I picked biology initially due to like concerns over just living on this planet Earth, I also began to realize that I don't think I would have made it very far in psychology because I think the chaos of it would have really gotten to me, which I think is kind of an interesting thing to think about in retrospect.
I mean, I do think like one of the reasons that we both had such a weird time discussing this is because there's that spectrum of like high external validity. Does this reflect the world and say like, does this generalize to other populations and species? And then high internal validity, which is, is very, very well controlled. And I just think as disciplines, like biology leans way more internal validity, and psychology definitely leans more in the direction of external validity, kind of by necessity, I would say. So it is just a very different way of approaching what we're doing, and I think some of that is due to the type of questions that we're asking.
Yeah, I think the reason why I'm not like crapping on your discipline, right? I'm expressing actually some appreciation for the people that choose to do your discipline. And this is going to sound like really backhanded, but I mean it genuinely. And the most flattering way that I can put it is that psychologists are battling the internal the intrinsic chaos of the human experience. And I think that's
that's very validating.
That's very brave, and that's why I don't want this to sound backhanded, because it's absolutely not. Like, I couldn't do it because it would drive me crazy. Like, trying to make...
I couldn't do what you're doing. It would drive me crazy because I would do the study, right? And I'd spend all this money and time on it. And in terms of generalizability, I wouldn't know if I would ever see this outside of the very specific circumstance I observed it in, right?
I think that's what sort of informs our perspectives on our disciplines and informs our preference for our disciplines, because actually, in terms of external validity, I've just kind of made my peace that someone's going to disprove this in humans later.
Yeah, and in terms of internal validity, I've kind of made my peace that, you know what, and especially with context being in there as well, I've made my peace that even if what I've found is a real effect, it probably won't replicate in another 20 years. Context has changed.
Science is hard, y'all.
Yeah.
And that's not to say that you can't have externally valid biology studies, right? That's what clinical trials are, essentially, right? Like giving drugs to people and figuring out if they work. Some of the best science comes out of that. Vaccine science, like science that is related to health in terms of just predicting, or not predicting, but gathering data on health outcome. Those are pretty good. They're done in humans. They're numerically rigorous. You can do them large scale. You can do them controlled, double-blinded, whatever you need to. Those are excellent studies. But in terms of raw mechanistic biology, the kind of stuff that I like, maybe it's only relevant in the fruit fly, or maybe it's only relevant in the mouse. Hard to know, but I'm okay with that. Because on a grant application, you can say that this may happen in a human, but that's up for the human scientist to figure out. That's not my job. I'm pushing the problem in the future.
Yeah. Well, we just keep pushing things off on each other's disciplines. Like, there may be a neural aspect of this, and then we just say, but that's not my problem.
That's true. We're like playing volleyball.
Pretty much. It's the problem, I think, is that when it comes to different disciplines like biology and psychology, we're playing volleyball, but we're in different courts.
That's pretty funny. Yeah, I think that's pretty accurate.
Yeah.
That's pretty good. All right. So, that's the end of that presentation. So let me go back to the title slide. Yeah, we made it to the end, y'all.
We did it.
Let me just reshare this. Instead of tabbing through all 60 of these monster slides, I will just redo this from the beginning. And we can chat about how people felt about the paper for a little bit, how you felt about the paper, if there are thoughts that you want to get at, or the experience of collabing here. That might also be helpful in case there are other VTubers who watch this VOD or in the chat who might want to think about coming on the Phylum channel to discuss some biology. So, how did the experience go for you?
I enjoyed it a lot. It was really, I learned a lot, both about VTubing, and I loved learning more about biology. And I did end up refreshing myself on things like principal component analysis stuff. So it was a very educational, and you are a wonderful collaborator. I couldn't do better. It's been a great time.
Thank you so much. And again, I'm always very surprised as to how hard my collab members work, because going out and scraping the CDC for data is just an insane move, but it's cool that you attempted. But yeah, humans are freaking messy.
Yeah.
So we have a little space now to talk a little bit about whatever. Chat. We'll take questions in terms of questions from the chat. I do want to emphasize I'm not I've made it very clear, I think, in multiple points of the paper that I'm not a microbiome expert. My perspective is one of someone who has a pretty good core of molecular biology understanding, but is presenting data as I would in a journal club, right? Where I'm not claiming to be the expert. I'm only claiming to be a guy that read the paper.
And when it comes to content, I am less than useless. I can talk about the stats, but the theory, I'm
Yeah,
useless there.
the stats is interesting too. Again, that's because in my journal clubs, I tend to just gloss over it because I just assume that they do it correctly. Because even when I was doing journal clubs for real in graduate school, I never really picked on their statistics. There were occasionally times when I'd be like, the n seems kind of low, just on an intuition from working in a similar system, but no real like hard line basis of, no strong conviction either way. Oh, can we talk a little bit about power,
Power.
if
Sure.
you can, for me and for the audience? So let me explain to you how I was taught power in terms of how we should apply this principle. So something that Reddit nerds love to get mad about is having a good sample size, and oftentimes in the comment section, again, you will hear them basically accusing the authors of having too low of a sample size, which is just like level 1 scientific criticism, right? Like, it doesn't take hard thinking to dismiss a paper outright because they have low sample size. Well, level 2 would be to think about power. And so power will describe the number of n's sample size that you would need for a certain effect to show itself to be real or not. And so I thought, cool, I like that. I don't want to think too hard about it. I'll just do the power formula and see how many n's I need, and then I'll just get those n's.
Mm-hmm.
So you had pointed out to me something interesting about power, something that is maybe led me to be quite uncomfortable with the idea of statistics as a whole. Kind of shook me to the core in terms of like, what even is statistics? I had an existential crisis. What are numbers? What is significance? Started like questioning myself on all these fronts. So from someone with a lot more statistical knowledge that I have, can you explain what power is technically?
Power is your ability to detect an effect assuming that it actually exists. So in psychology, we typically go for 80% power, which means, let's say,
that's very
assuming
anime.
that I know, right? I think I actually use Star Wars gifs like, I love power, or whatever, when I talk about it in class. But assuming that my independent variable has an effect on the dependent variable, there is an 80% chance that I will be able to find that effect, is typically what we would consider adequate power. So yeah, the power is the likelihood you will find enough that the effect is actually there.
Yes. Yeah. So there's a formula for power, so you can actually plug in some of your preliminary data to get a sense for how many n's you would need given a level of power. And what you had pointed out to me is that this is a very circular system where you need preliminary data to determine the power, but how do you know you had enough preliminary data to have enough to determine if the power test is useful? Like, there's this circular argument that I didn't really realize was intrinsic to the system, and then that's when I started to spiral out. I was like, how do I know I had enough initial sample size to generate the preliminary data to get an appropriate power? Like, it all seems kind of arbitrary, isn't it?
It is. The power analysis is a very tricky thing. One thing, there was a journal that I tried to publish in once that requires you to do something called a post hoc power analysis, which is after you've run this, based on the effect size you found, you determine if you had enough people to detect that effect. And as Marlon observed earlier, we mostly only publish studies where we did find an effect. So post hoc power analysis rejected the null is going to say that power to reject the null. And then if you're doing power analysis,
oh, sorry, your mic is cutting out again. This is so weird. It seems like when you start talking, it's fine, and then over time, the audio just kind of goes away. Let me try to see if like the filters are causing issues. So
yeah, maybe it's the settings that I've been having mic trouble today. So
I've turned off the filters, and hopefully that fixes things. It's a little weird.
Now I get to peek. This is what I've been waiting for.
Okay, I mean, I think it sounds a little better now.
Cool.
Yeah, so there's a circularity issue with power, right? Like, and so yeah, so there's a journal that you wanted to publish in that had you do another test to see if your power analysis was good.
Yeah, and so doing power before the fact is kind of circular because you need So effect size, how big of an impact things have, has a big impact on your power. So the size of your effect has a big impact on a power. So you need to have an estimate of how big your effect is going to be before you try and find the effect, which, you haven't found it yet. So how do you know how big it's going to be? And you can look at pre-existing, try and collect data. Yeah, let's see. Yeah, you can try and do those things, but at the end of the day, you're just kind of making your best guess. I changed the setting. We'll see if that does anything.
So far, it's good. Yeah.
Woohoo!
So that entire idea to me is so surprising, and I also feel like that explains why I have been describing statistics as an art and not really a science. Because ultimately, what even is significance? Like, what does it even mean for data to be significant? It's kind of an arbitrary cutoff where we agree as a society, as a community, what is considered to be meaningful and what isn't. Like, from its very inception, statistics is sort of socially defined. Would you say that that is okay to say?
I 100% agree. Like, that p is less than 0.05 as our benchmark for determining whether something's significant or not, and keeping it a strict benchmark. If it's even like 0.01 above this, it's not significant. If it's 0.01 below it, suddenly is meaningful. Even though that's not like a very big difference at all. We're just drawing arbitrary lines in the sand. It very much, we're disguising it behind a lot of math, still kind of making an arbitrary.
Yeah, and it can get kind of problematic, especially when different analyses cause you to like have to lower the p-value, and based off of what? Because it's a little more strict to do a lower p-value, and you need to be a little more strict when you are taking liberties with data or doing real analyses. But like, these are all sort of arguments from like vibes, right? Like, if you're doing multiple analyses, that's just like, you just have the vibe of like, you're going to find a false positive, which is like statistically true, technically, but like the choice to regulate the practice of doing post hoc correction is not like a thing that is rooted in math. It's a thing that's rooted in the psychology of researchers, right? Or the understanding of researchers of the statistical data. Like, it isn't like a hardcore science in the way that like, oh, objectively this is significant and objectively this is not significant. It's all like, we need to regulate how we do science in a way where we can define if something is worth pursuing or not. And that definition is kind of arbitrary, which makes rooting any of this in like logic very difficult, which is why like there's an interesting conversation in the Discord about whether or not stats is easy. And I think my answer was something to the effect of, stats is easy to learn but hard to master, because anybody can take stats 101 and learn what a t-test is and learn how to like look up a z table or whatever, and that's not that hard to do. But when you get into the field and you start using stats, you start to realize like, is this the right test? Should I be doing this? Am I gonna get canceled? Like all of these thoughts ran in my head when I think about when I was doing statistics in graduate school. Like, am I gonna get freaking canceled for doing this kind of test? Or like, is it okay to remove this outlier? Like, have I met the requirements? Like, it's all very difficult to do correctly, but it's not difficult to learn. I would say that has been my experience.
Yeah, I think it gets very difficult to learn after a certain point. Yeah, and then the implementation is a mess. Like, when you're at the level of t-tests and ANOVAs, I can make a decision tree to pick exactly which test you need, but once you start doing things that are more complicated, good luck. There's like 10 different things you can do.
Yeah, and something that has been an issue in biology, and this is especially relevant to this paper, is what counts as an N in a mouse? Is an N an individual mouse? Is an N all of the mice in a single cage, like a whole family? Because there are individuals that have correlation with their parental mice. Is a cage an N? Is a single mouse from a cage an N? Is each individual mouse that shares the same mother and father an N? Should they be an N? Because they have a correlation, they have mom and dad, they have that in common, so should they really be independent Ns? These are questions that biologists have been, I think there hasn't really been like a replication crisis level of reckoning when it comes to standardizing what an N should be in with regards to mouse studies. But maybe that's in the future, right? Because Myelin, when you pick an N or when you define an N, it's like a individual, and individuals are already quite unique, right?
Mm-hmm.
And so, from my understanding of psychology studies, N's are mostly just individual people who
It
have.
depends on what you're doing, but yeah, it can be individual people. It can be individual counties if your level of, it's whatever your level of analysis is.
Oh,
So
gotcha.
usually for us, that's people.
Gotcha.
Mhm.
But each individual, assuming that, oh god, this is complicated, right? Because population, and then you pick your sample from that population.
Yep.
But then, like, you can describe your population as university students, and then your sample is these students pulled from the psychology 101 class,
Uh-huh.
and they will have correlations in between them. A, they all go to the same university. B, they go to a university. C, they have like psychology as a required class because that is the major that they chose. So you have covariants that covary between these ends. And to me, that's a bit of a problem when it comes to that, because each individual N assumes that you are testing on someone who is unique. And I think that is more true of people than it is for mice, because if you've got identical mice, you know, you might be overestimating an effect because those are not really unique creatures. They are unique in the sense that they're autonomous, right? They move around on their own, but they're genetically identical, and they are like, they've got like the same mom and dad. So while like there are twin studies that, like, you know, they're genetically identical, but they're raised apart, or maybe they're raised in the same like way. But, you know, I think people have this inherent uniqueness that I think, for me personally, I'm more comfortable with calling them each their individual N. But for mice, because they are so similar, genetically similar, like, you can really obscure effect sizes by doing experiments on very, very, very identical mice. And you might think that's good. And in ways, I think that's fine. But if they co-vary in a specific way, that can create an effect where maybe there shouldn't be.
Oh man. So I want you to imagine me coming in here in like full salesman outfit. I brief and I open the briefcase and it says higher regression inside.
Higher what? Sorry, your
This
mic's
is what
cutting
you
again.
need. No! Hierarchical regression. Hierarchical regression would let you treat each mouse as an individual, but you can nest those mice within cages. Those cages with buildings and things like that. And that will help you deal with some of the covariances of a people who took the same class or mice that were raised in the same environment. Of course, then you have to deal with hierarchical regression, which is its own punishment.
You see, this is why it's useful talking to you, because it's like, yeah, your whole job is to deal with complicated people. And so, yeah, I guess I shouldn't be surprised that you already had an answer to that kind of thing.
I mean, it's something that we don't use very often because oftentimes we don't have things like that. But I absolutely have used it before, and a lot of people who study, say, like schools and things like that, they'll say all these students are in the same class, so we're going to nest each student within their class, next class with school, each school in the district. It gets messy, but there are ways to.
That is pretty cool. So there's a question in chat here. There's this thing that is still on my mind. Is the microbiome actually doing anything to the anxiety, or is it just the body getting stressed by the excess fat? We don't know from this paper. This is an early paper, 2014. I'm sure somebody has an answer out there. Since I'm more of like an immune system guy, my brain goes straight to, oh, high fat diet. If you have excess visceral fat, then that encourages inflammation. Inflammation can change behavior. That's where my brain would go to. But I don't necessarily know that that's the answer in the case of these particular mice.
Yeah, hard to say. My brain instantly went to social environment. Are the other mice reacting differently
Oh,
because of
to
this?
to the high fat diet mice. I mean, maybe.
Exactly.
Yeah,
Yeah,
they
but yeah.
could smell different, honestly, because mice, they don't really judge, because mice, their eyesight is really terrible. but they communicate and they interact through smell. So actually, high fat diet, they might smell different, which would be kind of interesting.
Yeah. Yeah, it's hard to say. The study probably didn't and probably even couldn't ask that question, but it is definitely an interesting question.
There are 400 citations for this paper, so somebody might have tackled it at some point.
Somewhere in there.
Yeah. Wouldn't it be practically significant since the difference from mice to humans is more genetically significant than mouse to mouse? Yeah, no, we're not really comparing between our specific fields in terms of like translating mice to human studies, more like how we treat what counts as an N versus how a psychologist treats what counts as an N. I think it's an interesting thought, because I do know that there's a paper out there that basically slaps biologists on the wrists for not being thoughtful about what counts as an N with regards to mice, which I think I agree with, because it felt like we were doing a lot of things that were just like flying from the seat of our pants, which is why I think we should take stats classes, like genuinely. I don't see why we don't. I don't see why we just learn from the people that came before us. Maybe it's not as big of an issue as I'm making out to be. But if you have like a very small lab where there aren't that many people to teach you, as I did, because when I joined my lab, the two most senior members and the only members of the lab, actually, they both graduated like the year I joined, which I didn't realize. So I was like the only lab member for like a year, which was really hard because I had to like do everything on my own. Including like learning how to do my own stats and everything. So I think that there should be a standardized statistics education in the curriculum for biology graduate students. Maybe you have one, but I certainly didn't.
You're preaching to the choir here. I think everybody should learn at least a little stats.
Just a little bit. Just a little bit.
A little bit.
Just a little bit. All right, we are creeping up on the 3-hour mark here.
Oh my gosh.
Which means that it is time for us to draw the stream to a conclusion. I will maybe extend the stream for a couple more minutes in case there are last minute comments, questions that the chat might have. But for now, is there anything that you would like to plug, Myelin?
I wish I had something to plug. Check out my statistics lectures. They're very nice. They're on hiatus right now. Come back.
And you do have a video, an interesting video about. Oh, can you pitch the video that you did about the BL and the brain scans?
Oh my gosh, yes. There was a paper going around Twitter that claimed that people who enjoy boys love things have neural differences from people who do not enjoy boys love, and people were freaking out about it, so I decided to look at it, and I did a long video discussing the methodological issues, the statistical issues, and some of the weird choices they had with how they reported the data, and some of the cool things that they found but didn't report in the main paper. I thought it was a really fun video. Yeah, I'd recommend it.
Definitely go check that out if you are a fan of BL and want to know what that may or may not imply about how your brain looks.
I won't spoil it for you, but I don't know how much you'll learn.
So please go check them out. Again, link in the description to their channel. And in terms of my plugs, I'm working my membranes off on a video about the immune system that should come out sometime at the end of this month. So if you haven't joined the Discord and you've made it to the end of the stream, then I will post a link to the Discord. Let me go ahead and do that. And I will like spam the ever-loving crap out of the Discord when that video comes out. Like, you will not be able to avoid hearing about how I have made this immune system video. I've worked so long on the video, and I think it asks a question that I myself have been long pondering, and I'm really, really excited to get it out for you all, and I hope that you will watch it when it comes out late October. All right. With that said, thank you all for joining me on this Friday night, a little off of my usual time slot here. But yeah, thank you for joining us and hearing us chit-chat about the microbiome. I hope you do nothing with this information, because you shouldn't, because this information is non-actionable. I don't know. maybe you exercise and eat a balanced diet. I can definitely recommend doing that. That's probably pretty good, unambiguously. But don't buy any supplement off of the information that you learned here. Yeah.
Yeah.
Being
Should I
said,
post the link?
yeah. Go post the BL video link. Myelin's channel link is in the description, but I don't have the actual link to the BL video in particular.
I don't know if links work in chat, but that is it.
All right.
Thank you so much for having me. This was a lot of fun.
Yeah, no worries. Thanks for coming on. Thanks for reading a paper outside of your comfort zone, and putting all the work that you did, and for the thumbnail, which looks great. Really, really
thank
awesome.
You too.
So yeah, bye y'all. Bye bye.
Bye.